Good morning, everybody. As you take your seats, I'd like to welcome you all to the second lecture, Dean's Lecture at the Michigan Medical School. Today we have the Dean's Lecture in the Bold Science Symposium, so we're very excited to have you all here. I just want to give a shout out to this event. We have over 300 registrations, including those who are online, so thank you for watching, and over 60 posters, so there's a lot of enthusiasm for this event. My name is Vicki Blanc. I'm an administrative director in the Office of Research, and on behalf of the Dean's Office and the Office of Research, thank you. I'd like to welcome Dr. Marshall Rangi, our Dean. Thank you for hosting this event. He will kick off the ceremonies today, and we'll be on our way. Thank you.
Thank you, Vicki, and thanks to all the people who worked on organizing this. This is going to be a great day, and with all the other things that we have on our mind these days, I think the opportunity to just think about what I believe is the most important area in health in the United States today, metabolic areas, and to have a true expert here is just a singular pleasure. President Ono couldn't be here today, but I would like to share a brief video that he made to introduce our symposium.
Good morning. I'm President Santa Ono, and it's a pleasure to welcome all of you to the second Dean's Lecture Symposium at the University of Michigan Medical School. In this, the school's 175th anniversary, I am so proud of your commitment to innovative education, pioneering research, and transformative care. I'm even more excited to see the future we'll create together. That's what makes this symposium so important. Today is a living, lasting tribute to the vision and compassionate commitment of Marshall Rangi. And I'd like to thank him again for his leadership as Dean of our medical school and service as Executive Vice President for Medical Affairs, a term that will be concluding this upcoming June. This symposium is also a testimony to all of you, to our shared dedication to saving and healing and lifting lives. It's not easy, nor are results always quick, much less certain. As a researcher in experimental medicine myself, I have the utmost respect for not just your brilliance, but your dedication and your determination. From our keynote speaker, Dan Skovronsky, we'll gain insight into the 100 years of intensive research into obesity that culminated in the development of Eli Lilly's new groundbreaking medicine. We'll also consider ways in which we may improve and accelerate this translational process, moving treatments and therapies more swiftly from the bench to the patient's bedside. This is a relentless commitment of Michigan medicine. It is also a vital element of our Vision 2034, our proactive dedication to human health and wellness and life-changing education. It goes to the heart of both impact areas and will be an integral part of our efforts and aspirations well into the future. So once again, welcome to the Dean's Lecture Symposium, and together, let's continue to define the future of medicine.
Well, hard to top that. And I hadn't seen it before. I would have edited out the part about me. But as President Ono mentioned, this is a reflection of our century and a half long interest in bringing science to medicine. And it's a tribute to all of you who are here. And let me just give my thanks also. So our Office of Research has put forth their strategic plan, Great Minds, Greater Discoveries, which is around trying to help improve what is already terrific, but our faculty's competitiveness and their ability to pursue major scientific questions in a rich and diverse environment that leads to discoveries, inspires new preventions, treatments, and cures. So Great Minds and Discoveries focuses on people, focuses on collaboration, focuses on bold science, clinical research, and infrastructure. And today we focus on bold science. metabolic research is incredibly timely, as I mentioned, and we've seen, I think, really incredible advances in therapies ranging from diabetes that began in diabetes to weight loss and beyond, as you'll hear from our keynote speaker today. Today's agenda will explore all of this complex subject, and we'll start with a few faculty members who will share their research, as well as being followed by our very special keynote address. And I'll save my comments for Dan at that time. We'll close the morning with a panel discussion about obesity and diabetes, which will feature several experts in the area. And this afternoon, we'll have a brief lightning round. I love the lightning rounds, to talk about ongoing research, followed by some faculty research insights and spotlights. And then we'll end with a poster session, both sides, and a reception. So I anticipate this being a great day. I'm very excited about it and very glad that you can be here. And at this point, I'd like to introduce Carl Jepson, Associate Dean for Research, who will introduce our faculty spotlights and the next part of the program. Thanks, Carl.
Thank you, Dr. Runge. Good morning and welcome to the Dean's Lecture and Symposium. Before we get started with our scientific talks, I'd first like to take a moment to thank the event organizers, and there are many. And this includes members in the Dean's Office, the Office of Research, and the Department of Communication. They have done a tremendous job in coordinating a lot of moving parts to make this day intellectually stimulating and very fun. So in addition, I'd also like to thank Dr. Martin Myers, and not to put him on the spot, but he played a tremendous role in planning this event. So he has done the work almost of an entire committee himself, okay, from helping to choose the day's theme, to assembling the agenda, and to the poster selection process. So we are very grateful for his contributions. In addition, several faculty members spent time reviewing the poster abstracts and helped select 5 lightning talks, as Marshall mentioned, and they'll be presented later today. So thanks to those members as well. And finally, thanks to all of our presenters who are sharing some exciting results of their research on metabolism. So please join me in a round of applause for our event planning and team and the presenters. So as you can see, we have a packed agenda. So we have 3 presenters for this next session. There'll be time for 1 or 2 questions after each of their talks. So the first presenter is Dr. Randy Seeley. He is the Henry King Ransom Professor of Surgery, Professor of Internal Medicine in the Division of Metabolism, Endocrinology, and Diabetes in the Medical School. Dr. Seeley's research is focused on the brain-gut axis and its role in the regulation of energy balance and metabolism. He and his lab work to understand the actions of GLP-1 agonists and identify the molecular underpinnings of the potent effects of bariatric surgery on weight and metabolism. The title of his presentation is To Be GIP or Not to Be GIP. That is the question. Dr. Seeley?
Good morning, everyone. Thank you to the organizers. And by the way, I'm really intimidated by the idea that you're going to get to see my words at the same time that I'm saying them. All right, so I first have to start with my disclosures, right? My lab works with a lot of different companies, right, that are working on advances in this field, including Eli Lilly. And you'll see data that in our work with Amgen at the end of this. All right, so one of the interesting things about all our advances around understanding how to treat obesity in particular has been the case that, in fact, that the one organ system that's been most important for us to understand this has actually ended up being the gut. So this is actually a review I wrote with scientists from Eli Lilly, pointing out, right, that the majority of what we can now do for patients really comes from an understanding of how the GI tract works, how it functions to be able to signal to other organ systems. So I will start by just pointing out something what's cool about the gut, right? It has 2 big jobs that are actually in opposition to one another. Right? So on one end, it has to be a barrier, right? It is the inside part of the tube that's keeping bad things from getting inside your body, right? But it can't be just a strict barrier, right? Because it actually also is in charge of absorbing all the nutrients, calories, water, all those things that you need, right? So that back and forth between balancing those 2 things is a really important part of what the GI tract does. And again, in omnivores like ourselves, it has to change over time. It can't be constant, right? So in fact, the gut has to change over time, right? And we call that enteroplasticity, right? The idea that the gut has to adapt to the situation that it finds itself in order to be able to do these two jobs. And one of the things that's really interesting about the gut is, again, it has its own brain. We call it the enteric nervous system. It has its own immune system. It has its own endocrine system. For any of the endocrinologists here, I hate to break it to you, but the gastroenterologist got all the good hormones, right? It's a barrier, as I talked about, and it has regenerative capacity, right? You're turning over your entire GI tract every 7 days, right? So again, that leaves its ability to be plastic, right? Because it's turning over all the time. It can be a very different gut 7 days from now than it is today. So again, we worry about the gut-brain axis, and you can imagine the importance of this inner part of our tube, right? And the health of it is incredibly important to the entire organism, which means that your brain has to know a lot about what's going on inside of it. And it has to be able to direct it to change in different times, right? So again, that connection both to the gut and from the gut turn out to be really important in understanding how it is that we might treat metabolic disease. The most effective tools we have are a direct manipulation of the GI tract. So these are two various flavors of bariatric surgery. I won't bore you with the details of these because it's not relevant today. But again, these are still the most effective things that we have. The drugs that I'm going to talk about are getting very close to being able to compete with bariatric surgery, but it's still the most effective thing we have. And the question is how, right? Why is it that these things work so well? And again, it becomes very obvious in clinical studies. This is a comparison at the Cleveland Clinic between folks getting medical treatment for their diabetes versus getting bariatric surgery. And you can see that both bariatric surgeries are much more effective in producing weight loss and much more effective in lowering people's blood glucose. So the point here, right, is that these are incredibly effective. And I'm now going to show you no more human data, right? I'm going to show you rodent data. And it turns out that rodent data is actually really important when we're talking about bariatric surgery because you cannot do a double-blinded randomized control study for surgery. Nobody wants their surgeon to not know what surgery they're doing while they're doing it, right? And the patients themselves know that they have trocar holes, right? Things happen to them, right? So there's no way for them to not know. So when you look at the data like this, what you're seeing is the confluence of the biological change we did by changing their GI tract, but also what they think is supposed to happen, right? And what I'm going to show you in the rodents, right, is that they don't get any instructions about what they're supposed to do. We do not have a rat registered dietitian telling them what they're supposed to eat afterwards, right? So what you see is purely the biological effect of the surgery, right? So this turns out to be rat. I'll show you mouse here in a minute, right? But what you see here are animals before surgery, right, on a high-fat diet versus animals that are maintained on a low-fat diet. This group of animals is now going to get vertical sleeve gastrectomy. And when they do, what you can see is that 34 days later, they've lost 50% of their body fat. They've lost all the excess body fat that accrued from being on that high-fat diet. And more importantly, they're still on the high-fat diet. They didn't change their lifestyle to achieve this particular outcome. Now the question is, how do they lose that weight? And the answer is mostly about food intake. So what you see in the aftermath of the surgery, right, is in that bottom line, are animals that reduce their food intake. But the most important part about this slide is that they don't reduce their food intake forever, right? They go back to eating what they did before. And that's not a physical adaptation. If I take the stomachs out on day 17 and look at it compared to day 8, those stomachs look exactly the same. It's not because I've dilated the stomach somehow, right? So the question becomes like, what is it happening here, right? Now, I'd make the argument, what happens here, right, is that the animal chooses to go back to eating what it did before because it's lost the weight that it sees as appropriate for the change in physiology that we produced in the animal by doing these surgical procedures. I'll try to prove that to you. So this is an experiment where we've taken animals, given them vertical sleeve gastrectomy or a variety of other control groups, okay? Now, what we're gonna do after the animal's lost the weight is we're now gonna force it to lose more weight. We're gonna put it on a diet, right? So we're gonna food restrict the animal and you can see that they're gonna lose weight. Now, what happens in a normal animal, right, if you food restrict it, is that it's hungry and it eats more and it regains the weight that it lost, right? That is the defensive body weight. Now, the question becomes, in the case of an animal with vertical sleeve gastrectomy, will it do the same? So I'm going to change the axis here so you can see, put all the animals starting at the same place. And what you see is that when you give an animal vertical sleeve gastrectomy, we drag their weight further down and give them back the food again, they overeat and regain the lost weight in a manner that is absolutely identical to the other groups. So the important thing here, we didn't break the animal, right? It's not, we haven't screwed up the animal's GI tract so that it just happens to lose weight because it can't gain weight. It's because the animal, we've changed the set point. We've changed what the animal thinks is the right place to be. And so the animal will work to get back to there, right? And I'll just show you, the way they do that is to be able to eat more, right? So this is the control group, right? So animals, after that food restriction, you can see in that orange bar, right? They overeat in order to regain the lost weight. 2 more control groups do the same thing. And the animals had vertical sleeve gastrectomy, overeat to be able to get back, right? They defend a weight. They defend a lower weight, right? But they defend it. And the important point is, and if you look in those pink bars, they look all the same, right? The animals are eating the same amount after they, but the animal who in the VSG group is 50% less body fat. So from our outside perspective, right, that animal's underweight. But the animal doesn't act that way, right? Because it's not overeating. But it's not because it can't overeat, because now if I drag the animal further down, it shows you that it's perfectly capable of eating more calories if it wants to. So the point about all of that is you cannot do that without imagining that you're changing the brain, right? There is no version of the animal changing its relationship with the food environment like that without having changed things that are going on in the brain. And I think the bottom line of all the things I'm going to tell you, right, is that the central nervous system is absolutely critical to the changes that we do using these GI tricks, right, in order to be able to produce a change in weight and a change in that set point. Now, how do we begin to dissect molecularly what it is that we did to the GI tract that results in a change in the brain, okay? So that's a bunch of what we do in my lab, is trying to understand that. One of the things that changes after surgery is the barrier function of the gut, right? It actually gets better, and I'll show you that in a minute. And the question is that change in barrier function an important part that contributes to the success of the surgery? So most of you probably appreciate that part of the way the gut produces a barrier, right, is one is physical tight junctions, right, to make things hard to sneak out of the gut. It's also got mucus production, right, which sort of is spackling along there, right, to make it hard for things to sneak out. But there's also a chemical part of this barrier, right? In various cell types, you make antimicrobial peptides that are actually really important to keep the bacteria that are in your gut and not let them leak out into the rest of the system. One of those is an antimicrobial peptide called reg3 gamma. And if you do bulk seek for what happened after bariatric surgery, right, and look at the change in the transcriptome, the thing that leaps out at you when you look at the GI tract, right, is an increase in the transcription of these antimicrobial peptides, and the one that is increased the most is reg3 gamma. So now how do you know whether that increase in reg3 gamma matters or not, right? So the crux of what we do, right, is to build an animal model where it lacks reg3 gamma and ask whether it responds to surgery, right? If this increase in reg3 gamma is important for why surgery works, then that animal should be less responsive to the surgery. So again, we're going to take wild types and knockouts and do vertical sleeve gastrectomy. First thing here, right, again, is the improvement in barrier function, right? So we use something called Fitzy dextran. We put it into the gavage of the animal, and we just measure how much of it gets out, right? Again, the more that gets out, the worse the barrier function is. And you can see in VSG animals, right, that barrier actually gets better as a function of having changed that GI tract. And it turns out all of that depends on reg3 gamma, right? So without reg3 gamma, you don't see any improvements in that barrier function. How about the other things, right? So here's body weight. You can see a reduction in fat mass that happens in the wild type animals does not occur in the animals that lack reg3 gamma. The other major thing that surgery does is to improve glucose. This is fasting glucose, you can see improve after bariatric surgery, and all of that effect goes away in the animals who lack reg3 gamma. So the important point here, right, is that reg3 gamma is necessary to the benefits of vertical sleeve gastrectomy. It's important to remember that the surgeons who did these studies made the stomachs just as small in the knockouts as they did in the wild types, right? But a small stomach isn't enough. It isn't sufficient to actually drive the benefit if you do not have signaling systems like reg3 gamma. Okay, so why does this matter, right? So all of this has changed my view about exactly how it is that this system works. We know that both type 2 diabetes and obesity are complex interactions between the environment we live in and the genetics we bring to the table. What I think is less appreciated is that how is it that that environment is impacting the organism? And at least one major way it is doing so is through the GI tract, right? bad diets, environmental insult, environmental toxins, all will create gut dysfunction. In an animal, right, in an organism, right, that is predisposed genetically, right, to have dysfunction, that creates pressure on metabolic organs, which creates metabolic deterioration, right? And that metabolic deterioration, turns out it adds to more gut dysfunction, right? High blood glucose levels make the barrier function of the gut worse. So you can see that that the GI tract is part of a negative feedback loop, right, that actually contributes to the ongoing deterioration of the disease. And the point here, right, is that we're trying to understand how it is that the gut does that in order to be able to fix that in some way, right, that produces better outcomes. Now, everybody in this audience knows, right, that bariatric surgery is not the only thing that matters, and that we have gotten so much better, right, at being able to use other sorts of treatment strategies to treat both individuals with type 2 diabetes and obesity. And the first of these comes from doing analogs of various gut hormones. This is an analog of a gut hormone called GLP-1. This long-acting analog produces reductions in food intake and body weight. And then you can see here in humans, right, in clinical trials of about 16%. And what I want to show you, right, is that this has some commonalities to what happens after bariatric surgery. So first of all, when you do these mouse models, what you see is a reduction in food intake that drives the majority of that response in terms of decreased body weight. But again, that effect doesn't go on forever. It goes away, right? And some people might think, well, maybe that's tachyphylaxis, right? That the drug has stopped working. And I would argue that is not what's going on here at all, okay? Because when you look at body weight, the body weight effect stays, right? The animal has achieved a weight loss. Now, once that weight loss is appropriate to what the animal sees, and now with the drug on board, the animal goes back to eating what it did before, right? And this is true in humans as well, right? So again, one of the interesting things, right, is again, after you see the reduction in body weight, if you take the drug away, what happens? Well, people start regaining that lost weight. Again, that makes sense, right? So again, it's not tachyphylaxis in the usual sense, right? Because what happens here, right, is that when we take the drug away, you can see here in the animal models, what they immediately start doing is overeating, right? They don't go back to eating where they were. They start now overeating in order to make up the lost ground, right, to get back to the weight that they now think is appropriate, right? Now, I would argue that this is the brain, okay? That what happens here, right, to change that weight has to have actions in the central nervous system directly. So one of the things that we did was to build an animal model where we either knocked GLP-1 receptors out of peripheral neurons or central neurons, right? And what you see in the bottom two lines, right, are wild-type animals or animals who had GLP-1 deficiency in peripheral nerves. Doesn't matter, right? You lose weight in response to this GLP-1 agonist. But if you get rid of the GLP-1 receptors out of the brain, all of that effect goes away, right? So again, what matters is the ability for these drugs to act directly on GLP-1 receptors in the central nervous system. So this is in here to remind me to tell you about one of the other things that happens with these drugs, right? Which is that many patients experience nausea and or vomiting at the beginning of starting to take these drugs. And we want to be able to understand that and be able to figure out how it is that that occurs. And that requires a real problem, right? Which is, how do I know when the animal is sick, right? So all of you have actually experienced how we do this, and it's called a conditioned taste aversion. Mine is with tequila, okay? So first time I had tequila, it was an unusual flavor. Several hours later, I was violently ill. We don't have to talk about the role that dose might have played, but to this day, right, I cannot stand the sight of tequila. I know cognitively that if I took a smaller dose than I did that previous night, that I would be okay. But I still can't touch it, right? And the way we do that in an animal is to give it a novel flavor, we usually use saccharin, give it something that might make it sick, and then come back several days later and ask the animal, do you want to drink the saccharin or not, right? And what you see here is that if you use lithium chloride, something that we know makes people sick, you can see that it reduces the ability of that animal to want to consume that saccharin solution, and so does a GLP-1 agonist. And it turns out, again, that does not rely on GLP-1 receptors in the periphery, but it does rely on GLP-1 receptors in the brain, okay? So why do I bore you with that? I bore you with that because it's a feeling that people have of what something that's happening in their GI tract, but it's not happening in their GI tract. It's happening in their brain. Right? Okay. I'm going to skip this part. All right. So the magic of what we've been able to do now, right, is now rather than stopping at just giving people one gut hormone, it's the ability to be able to do more than one. And I am sure our speaker is going to tell us a lot about this. But the important part for you to remember, right, is that what Lilly has done here, right, is to not give two drugs at once, right, by just attaching them in some way. It's to cleverly mix the peptide sequences between GLP-1 and another gut hormone called GIP to be able to turn on both receptors at the same time. And when you do that, you can get more weight loss, right? Seems very clear. So now the question is, how does that work, right? And it's even more difficult because I can tell you I was part of arguments for this over 10 years of arguing about whether it was the right idea to turn on the GIP receptor or to turn off the GIP receptor. And the argument about turning off the GIP receptor looks right here. The genetic models where you get rid of GIP receptors makes for a skinnier animal. It's not just mouse genetics, right? It is human genetics, right, that leads you to the conclusion, right, that in humans with relative loss of function of GIP, that they end up to be protected from weight gain. So again, what's interesting about this field is, well, Lilly made a bet and has successfully prosecuted a GLP-1 GIP agonist. Other companies took the opposite side of the bet, right? This is actually a molecule from Amgen in which they conjugated an antibody that is a GIP antagonist to a GLP-1 agonist. And the beauty of this, right, is that you get a clear synergistic effect, right? That taking the two things together gives you more weight loss, right, than either of the components alone, right? If you're not confused, you're not paying attention, okay? It doesn't make sense. So we have spent a lot of time trying to square that circle, and I'm going to give you just a tiny bit of how we try to do that. So what we have done is to build GIP receptor knockouts, either whole body, right, and you can see here, right, that the animals are leaner, just as have been replicated by others. And then we knock GIP receptors either out of the glutamatergic neurons, right, the excitatory neurons, or the inhibitory neurons. And it turns out, again, the brain is what matters, right? Because when we knock GIP receptors out of the glutamatergic neurons, the GABAergic neurons, you recapitulate the phenotype of the entire body. Now the question is, what's the response to these drugs? So the confusing part here, right, is that when we take a GIP receptor knockout and we give it tirzepatide, that animal's response is actually better. Again, Randy, you just told me that taking the two things together causes more weight loss, but if I get rid of the signaling on one of them, the response gets better. Well, that's all true, but it turns out it's a bit of a trick here, right? Because tirzepatide is not a very good mouse GIP agonist. So what you see mostly in the mouse for tirzepatide has to do with what it is doing on the GLP-1 side. So what we've done is try to do experiments where we build a mouse tirzepatide, meaning we give tirzepatide and then we give a mouse GIP agonist with it in order to create the signaling of both. And what you see here is that tirzepatide causes weight loss. In the knockout, in the brain knockout, right, of GIP receptor, you now see that it is better, right? So just like in the whole body, right? Again, tirzepatide is more effective, right? So GLP-1 agonist is more effective in the context of a lack of GIP signaling. Again, whether we didn't do it pharmacology here, right? We did it with genetics. What's interesting is that if we now give tirzepatide with a GIP agonist, it's better, right? Again, adding GIP on, right, actually does make a molecule that causes more weight loss. And finally, again, if I now do that in the context of the knockout, you can see that, in fact, I can get rid of all of that extra effect, right? So again, the GIP receptor matters, but it matters in both directions, and it's the same population that matters in both directions, right? That is super confusing. Right? And I think Paula will address some of this later. Again, so we're trying to square that circle. Now, I'll skip that. What we're going to end up with here, right, is again that what matters to changing weight has to do with changing that set point by changing actions in the brain, right? And that's what's common between both surgical approaches and pharmacological approaches. And I'll end with pointing out that I didn't do any of the work you just saw, right? It's a talented group of folks here at the University of Michigan. So thank you so much for your time and your attention.
Are there any questions for Dr. Seeley? Okay.
So, Randy, thank you very much. What a great presentation. Thank you very much. That was a great presentation. I remain confused about the GI antagonist story. And I'm sure that's something you're actively investigating. Do you have any kind of insights into that? I mean, that was totally new news to me.
So I think, again, first, right, if you're not confused, you're not paying attention. It turns out that everybody was right? All the people who are arguing for a GIP agonist, they were right? Tirzepatide is a very good clinical asset and effective, right? Amgen's approach works as well, right? They are to phase 2 trials, right? And show, again, long-lasting effects in human as well. Again, so now again, getting down to the details of that will matter to the way future therapies get constructed, right? Which side of that equation do you want to be on, agonism versus antagonism? Again, I don't think there's a clear answer to, they both work, right? At the end of the day, right? And I think it speaks to one thing that's really important is none of us are as smart as we think we are, right? Lots of highfalutin arguments were made about GIP agonism versus antagonism. And again, both end up working, right? Everybody had a piece of the story correct at the end of the day. Great. Thank you guys so much. I appreciate it.
Thank you, Dr. Seeley. That was an amazing presentation. So our next speaker is Dr. Martin Myers. He is the Maryland H. Vincent Professor of Diabetes Research, Director of the Michigan Diabetes Initiative, and Professor of Internal Medicine, Division of Metabolism, Endocrinology, and Diabetes. Dr. Myers' research focuses on the processes that enable the body to respond normally to insulin and how problems in these pathways contribute to the development of insulin resistance and diabetes. Dr. Myers.
Okay, so I'm going to start by really telling you how we got involved in the research that we're doing probably about 10 years ago. And this was a series of conversations with Randy Seeley, actually, who, in addition to being a researcher who does interesting stuff, is actually trained as a psychologist. So as you'll see, was able to explain to me the fallacy of my thinking when we started out. So we started with the question of whether it would be possible to separate the suppression of feeding from nausea. And the way I started thinking about it, again, just based on my own personal experience, is that you eat a little bit of something, at some point in time you start to feel full, you eat more, you get really full, and then if you keep on eating, at some point in time you get so full that you get sick, right? And this suggested to me, what it may suggest to you as well, that this model is that there's a single response system that sort of links the gut to the brain. And if you push on the system a little bit, you get full. But if you push on the system really, really, really hard, then you're going to get sick. So again, this is the single circuit model. And this would predict that we actually can't we can't separate appetite from nausea. Well, what Randy actually tried to convince me of, and I think ultimately did convince me of, is that model doesn't make any sense at all if you think about what the gut needs to tell the brain, right? So one thing that it needs to tell the brain is if you eat food, one of the donuts or something that's back there, you need to change your behavior and you need to change your gut physiology to take advantage of that, right? So you need to change your gut physiology so you absorb the calories that you're taking. You need to, at some point in time, become satiated so that you don't eat too many calories. And the other thing you need to do is that you need to remember that that was good and that you should circle back at some point in time. And if you're presented with that food again, you should eat it again. Now, on the other hand, if you eat something that's toxic or irritating, you also need to stop eating, right? Because you want to stop ingesting whatever the toxin is. But you don't want to absorb that, right? You have to have an entirely different response, which is to expel the noxious substance that you've eaten. So that's nausea, vomiting. And then as Randy pointed out, you also need to remember to never eat that again. You need to form a conditioned taste aversion. So the point here is that it doesn't make sense for a single system that communicates from the gut to the brain to mediate both satiation and aversion, right? Nausea and vomiting. So at that point in time, we started thinking about how we might be able to parse these two systems. And here, I'm just going to give you a little bit of background into how the hindbrain is structured and ways to think about how we might tear apart the two systems. So the gut communicates to the brain through a couple of different mechanisms. One is that it secretes gut peptides, like you just heard about from Randy, that go through the circulation. And they bind to neurons in the area postrema, which lives in the hindbrain outside of the blood-brain barrier. The neurons in the area postrema project into a place called the nucleus of the solitary tract, which does get some information from circulating peptides as well. And then there are gut sensory, vagal sensory neurons that innervate the gut and bring that information also into the nucleus of the solitary tract. Now, when we started this, we knew that the area postrema was probably going to be an important place for mediating these aversive signals. The area postrema actually is known as the vomit center of the brain. And we knew that there were two flavors of neurons in the area postrema that were important for, that were able to turn on anorexia and aversion. Those contain GLP-1 receptor and then the receptor for a peptide called GDF15 called Goforth. And we also knew that one of the main reasons or the main mechanisms by which they mediate these aversive responses is by turning on CGRP neurons in the parabrachial nucleus. We also knew that there was a set of neurons in the NTS that contain CCK, and these also turn on those aversive neurons in the parabrachial nucleus and also turn on aversive signaling. But the question is, where's the set of neurons that mediates the non-aversive suppression of food intake? And so that's what I'm going to spend the rest of this, or most of the rest of this, talking about. The other thing that I'm going to point out is that at the time that we started doing this, the idea that I think everybody held in their heads is that the hypothalamus is really important for the long-term control of food intake, and that the brainstem just modulates things like meal size. So if you push on the brainstem, your meal size might go down, but then the hypothalamus will tell you to start eating more meals, and meal frequency will go up, and you won't actually get a long-term suppression of food intake. So I'm not going to go through all of the ins and outs of it, but at some point in time, we decided that there were two populations of neurons in the nucleus of the solitary tract that were likely to mediate the non-aversive suppression of food intake. These are marked by calcitonin receptor and leptin receptor. So one of the tricks that we use for manipulating these neurons is something called a DREAD, designer receptor exclusively activated by designer drugs. This is a human muscarinic receptor that's been mutated in vitro so that it no longer binds acetylcholine, but only binds this otherwise inert artificial ligand called clozapine N oxide. And so what that allows us to do by packaging it into Cre-inducible AAVs is to put this stuff in the neurons that we care about, in our case in the nucleus of the solitary tract, so that we can turn them on at the drop of a hat just by injecting clozapine N oxide. So this is that experiment. We've taken 4 flavors of mice here, just plain old wild type animals, mice that contain leptin receptor Cre, mice that contain calcitonin receptor Cre, and mice that contain both Cres. We put the DREAD in the NTS, we turn on with CNO, and you can see that turning on either or both of these really suppresses food intake at the onset of the dark cycle, turns down refeeding following a fast, and even if you give them tasty high-fat diet, turns things down like crazy. And I'll also point out that when you turn on both populations at the same time, food intake goes down to 0. And in fact, if you do that over a longer term and keep the neurons on for a couple days, you can see that in the doubles, right, where we're turning on both populations of neurons, food intake actually stays at 0 for 24 hours and then bounces a little bit back that next day. But the point here is that by turning both sets of neurons on, we can push so hard on the system that food intake goes to 0 and stays at 0. So what happens to aversive signaling. And again, as Randy said, you can't actually get rodents to vomit, or at least mice and rats to vomit. So we have to use this trick called a conditioned taste aversion, where we pair a stimulus with, in this case, their first exposure to saccharin in water. And I'm just gonna switch through that and show you, this is what happens if you turn on the neurons with, you turn on the CCK neurons, which I already told you are aversive. This is what happens if you turn on the calcitonin receptor neurons alone. So again, CCK neurons aversive, calcitonin receptor neurons not aversive. And in fact, if you turn on both the calcitonin receptor and the leptin receptor neurons at the same time, even though you're pushing so hard on the system that food intake goes essentially to zero, no aversive signaling at all. So there are, in fact, two different populations or two different circuits in the brain that can suppress food intake. The other thing I'll show you is that if we take these leptin receptor and calcitonin receptor neurons and instead of turning them on, we chronically turn them off by expressing tetanus toxin in them. Not much happens when you feed the animals chow, but the minute you switch them over to a high-fat diet, they start eating more, and you can see their body weight goes up like crazy. So over the ensuing 5 or 6 weeks, these animals gain 20 grams of body weight, right? About more than twice what the controls would. So the endogenous activity of these neurons is also important for the ongoing suppression or the ongoing maintenance of body weight. All right, so what I've shown you is that we've got two populations of neurons in the NTS, calcitonin receptor, leptin receptor, that are capable of mediating satiation without aversion, and that also contribute to the physiologic long-term suppression of body weight. Now, what about those CCK neurons? Do they actually contribute to long-term energy balance as well? So again, we put the tetanus toxin in the CCK neurons in the nucleus of the solitary tract, switched them onto chow and then onto high-fat diet, and you can see that turning these neurons off also increases food intake on a high-fat diet, which suggests, obviously, that these neurons are also involved in the long-term suppression of food intake and body weight. Now, the problem with that is that we also know that if you turn off the CGRP neurons, the sine qua non of aversive signaling, you don't change energy balance, right? Nothing happens at all other than changes in meal size and meal frequency, like I mentioned a few minutes ago. So what that suggests is that the CCK neurons actually have a projection that goes through this aversive pathway, but they must have a projection that does non-aversive signaling as well. And in fact, relatively recently, Laura Heisler in the UK showed that the leptin receptor neurons overlap with the CCK neurons, which suggested to us that the CCK neurons may not be a pure population. And when you think about it, that could also be true of the calcitonin receptor neurons or the leptin receptor neurons. So we set up a collaboration with a guy named Tuna Peres at the University of Copenhagen. And what Tuna did was essentially dissect the dorsal vagal complex, the area postrema, the NTS, subjected it to single nucleus RNA sequencing, and then came up with one of these UMAP plots where all of the neurons are clustered into neurons that have gene expression patterns that are similar to them. And he came up with a bunch of them. We were able to help him assign them to the area postrema, the NTS, or the dorsal motor nucleus of the vagus. And this is just looking at some of the things, some of the markers that we care about. So the first thing you can see here is that there are two populations of calcitonin receptor neurons. Only one of them is in the NTS, so that's actually a pretty good marker. That's a relatively pure population. If we look at the leptin receptor, it turns out there are two populations of leptin receptor neurons in the NTS, and so that's not a great marker. And if we look at the CCK, it actually three populations of neurons in the NTS, so that's really particularly dirty. The other thing I'll point out is that there are two populations of GLP-1 receptor expressing neurons, one in the AP and one in the NTS. So the map of all of the AP NTS neurons that we came up with as a consequence of this kind of looks like this. So we've got these, what he calls GLU11 calcitonin receptor neurons that live in the NTS. These, as I've shown you, mediate the non-aversive suppression of food intake and contribute to long-term energy balance. There's one population of leptin receptor neurons that does the same thing. These actually express pre-proglucagon and make GLP-1 in the hindbrain. But then there's a second population of leptin receptor neurons back there that are involved in the control of respiration. And then there are two additional populations of neurons that contain CCK, and we think that one or both of these are probably involved in aversive signaling. I'm not going to talk about those today, but talk a little bit about where GLP-1 receptor agonists might mediate their effects, because one set of neurons contains GLP-1 receptor is in the NTS, and that's actually the same population of neurons as contains the calcitonin receptor. So we would predict that the NTS GLP-1 receptor neurons are involved in the non-aversive suppression of food intake and are also involved in the long-term maintenance of energy balance. And as I already told you, there's another population of neurons, of GLP-1 receptor neurons. They live in the AP, and we would expect that those are involved in the aversive suppression of food intake. So we set out to test that. And what I'm showing you here is, again, using those activating dreads, activating either GLP-1 receptor neurons in the AP, which is what we have on top here, or activating GLP-1 receptor neurons in the NTS. And you can see in both cases, when we add CNO, these neurons go on, food intake goes down, body weight goes down, we can keep it going down for several days. And as I told you, when we turn on the GLP-1 receptor neurons in the area postrema, we get a huge conditioned taste aversion. We do the same thing with the NTS GLP-1 receptor neurons, no conditioned taste aversion at all. Furthermore, when we do, again, the opposite experiment by putting tetanus toxin either in the AP GLP-1 receptor neurons or in the NTS GLP-1 receptor neurons, in the AP, nothing happens to the control of food intake or body weight. Right? There's no change on chow, there's no change on high-fat diet, but as you would expect based on the fact that those NTS GLP-1 receptor neurons map to the same population as the NTS calcitonin receptor neurons, as soon as we switch those animals onto the high-fat diet, they eat like crazy, gain a ton of weight. So that suggests that if we can make a magic GLP-1 receptor agonist that would just hit the NTS GLP-1 receptor neurons, that it would be the drug that you really ideally want, because you get the suppression of food intake, you get satiation, but you wouldn't have all of this nasty nausea and vomiting. So to test and see whether that was actually even theoretically possible, we took a GLP-1 receptor allele that was made by Randy Seeley and used that to turn GLP-1 receptor back on just in specific populations of neurons. So the way this allele works is it has what we call a stop flux cassette in it. So it's got a LOX-P flank transcription blocking cassette right in the middle of GLP-1 receptor. So in the state of nature, nothing gets expressed, but if you add Cree, that transcription blocking cassette comes back out and you get native GLP-1 receptor expression just in whatever population of neurons you're interested in. So I'm going to show you experiments now with 4 flavors of mice. One of them is these lock-stop-lox animals, which have no GLP-1 receptors anywhere. 1 wild-type animals. 1 set of animals where we've reactivated specifically in the area postrema, and 1 where we've reactivated specifically in the NTS. And when we look at the response of liraglutide in these animals, what you can see is that obviously the animals that have, oops, where's it going? The animals that have no GLP-1 receptor, no response. The animals that have wild-type GLP-1 receptor, food intake and body weight go down. AP GLP-1 receptor reactivation, food intake and body weight go down. NTS GLP-1 receptor reactivation, bupkis, which suggests that for whatever reason, these NTS GLP-1 receptor neurons just aren't able to get enough of the of GLP-1 receptor agonism going to do what we really want on those neurons. So what that means is that although the NTS GLP-1 receptor neurons are capable of mediating non-aversive satiation, and they are involved in the long-term maintenance of energy balance, for whatever reason, GLP-1 receptor agonists don't do anything on those neurons. Rather, most of what we see happening in response to GLP-1 receptor agonists is happening by these AP cells. Now, the last thing I want to show you is that we're interested not just in what circuits suppress food intake and mediate nausea, but GLP-1 receptor agonists do a lot of other interesting things, many of which seem to be clinically relevant, including the suppression of inflammation. So this is actually from a really nice paper from Dan Drucker about a year ago, showing that if he takes wild-type animals, gives them vehicle and then LPS, he gets this crazy cytokine storm. If he gives them exenatide, which is a GLP-1 receptor agonist, before he gives them the LPS, he suppresses the cytokine storm, brings it down by about 30 or 40%. And it turns out that all of that effect is mediated by GLP-1 receptors in the brain. So when he knocks GLP-1 receptor out of the brain using one or the other of these Cres, that anti-inflammatory effect goes away. So of course, we decided that we should throw our model in that too, and these are relatively preliminary data. But I'll just show you that in wild-type animals, again, we see the same thing that Dan Drucker did, which is that semaglutide can suppress the cytokine storm, that the semaglutide doesn't suppress the cytokine storm in the knockout animals, but in the animals where you reactivate GLP-1 receptor in the area postrema, you actually get suppression of the cytokine storm. So what we think is going on is GLP-1 receptor agonists are mediating most of their effects through the area postrema. Certainly their aversive effects, their anorectic effects, they're suppressing inflammation. I didn't show you this, but it turns out there's suppression of gastric emptying also happening through the AP. And we're very interested in some of the other things that are coming out of GLP-1 receptor agonists. And at that, I guess I'll stop. I thanked a few people as we were going along, but I need to specifically thank Warren Yakovich and Yi Wang, who did a bunch of the stuff with the GLP-1 receptor reactivated mice. Mark Padilla is doing the anti-inflammatory stuff. And of course, this is all a huge collaboration with Randy Seeley and Paula Goforth and Darlene Sandoval, et cetera. And I guess that's it. I'd be happy to take any questions.
Hi, Liz Veliotis. I was wondering, with these long-acting agonists, do you see a downregulation of the receptors? In which case, could that be a mechanism of how loss of function could be the equivalent of long-term agonist
So are you
effects?
talking about GLP-1 receptor?
GLP-1 receptor, yeah.
So you're asking maybe specifically about why the GLP-1 receptor neurons in the NTS aren't responding?
Well, I mean, these are long-acting agonists,
Yeah.
right? So biologically, you could have a downregulation, and
Yeah.
that would be equivalent of a knockout.
Yeah, so that is the case. In fact, I think that's a reasonable guess for what's going on with GIP agonism versus antagonism. We actually think that what's going on with the GLP-1 receptor neurons in the NTS is that they just don't have much GLP-1 receptor. And so we're in the process of doing that experiment by trying to crank up the amount of GLP-1 receptor they express and see whether we can hotwire them so that they actually would respond.
So these are terrific talks and they're amazing drugs, but it is really complicated. On top of which I'm sure there's genetic variation among the patients. How are you starting to address that?
Sorry, how are we starting to adjust genetic variation?
Yeah.
Well, to be glib, that's actually his job. But I mean, there are a lot of GWAS studies out there, and certainly GIP receptor is one of those things that comes up. And I would expect that, actually, that's an interesting question. Has anybody ever tried tirzepatide or looked at tirzepatide in people who have the GLP-1 receptor, like the GIP receptor? It's not really a knockdown, but the hypomorph. Yeah, that would be interesting. I don't know. Yeah.
Thanks for a great talk. I was thinking about that Drucker paper you showed with the LPS challenge and the anti-inflammatory effect in the brain, which was compelling. I think in that paper he showed that tirzepatide retained an anti-inflammatory effect in the GLP-1 knockout, suggesting GIP. Wondering if you're testing GIP specifically in those kinds of models as well.
So yeah, we are not testing GIP. Well, not in that way. So we've made a set of, it's very hard to manipulate the AP in the mouse just based on where it is and how big it is. So we've made a set of Cree rats that we can use to manipulate things in the AP. And so we are looking to see what happens when we manipulate AP GIP receptor neurons versus NTS GIP receptor neurons.
Time for one more question.
Thank you for that talk. Could you maybe tell us something broadly about whether or not you see any sex differences in some of these either main effects in terms of parsing apart the aversive versus non-aversive components, or some of these circuits?
So, I mean, the short answer is, we do actually look at both males and females in all of the assays. We haven't seen sex differences, but we're not really powered to look at sex differences. So I think it's entirely possible that there would be.
Can be very different in men and women, particularly during pregnancy. So maybe I was just thinking about opportunities there where there might be logical reason to think that some of those mechanisms might be very different.
Yeah, absolutely. All right, thank you.
Thank you, Dr. Myers. Our third speaker today is Dr. Paula Goforth, and she is the Assistant Professor of Pharmacology. Dr. Goforth's lab studies the neural control of energy balance and ingestive behavior. They are particularly interested in the cellular and molecular mechanisms that underlie the regulation of neuronal populations and circuitry that promote satiety or drive food intake. They also examine how disruptions in these physiological systems lead to or are altered by disease processes such as obesity, dysregulated feeding, and addiction. And the title of her talk is Incretin Regulation of Hindbrain Neurons.
Shorter. Okay, thank you for that. I also want to thank Randy and Martin for providing some nice background to the talk I'm about to give. My lab studies, as you said, neural control of energy balance. First, for disclosure, I also received some research support from Eli Lilly for some other research projects. We are particularly interested in the control of energy balance, and that includes the role of gut-brain communication in affecting satiety, such as meal timing and meal frequency. I hope that Martin has impressed upon you the integral role that the hindbrain, and in particular the dorsal vagal complex, plays in energy balance. The hindbrain and the DVC receives input from vagal afferents that signal visceral stimuli such as stretch and nutrient absorption. They also can respond to circulating factors and such as hormones that are released from the pancreas or adipocytes. For this talk, I am particularly interested in the role of GIP signaling in the hindbrain neurons in the dorsal vagal complex. And as Randy has already gone over, the GIP signaling and GIP receptor biology is very complex and contradictory, right? So we have the case where on one hand, GIP receptor agonism suppresses food intake. This is particularly evident in these unimolecular coagonists, such as tirzepatide. On the other hand, GIP receptor antagonism or loss of function of the receptor can also suppress food intake and acts as an anti-obesity agent. So one of the questions in my lab is, how can this be, right? What can we provide some insight of how GIP acts in the brain to result in these disparate effects? And very little is known about how GIP acts as receptor, the GIP receptor in the brain. Some studies have been done in islets or adipocytes, but very little has been done in brain. And for these studies, I'm particularly interested in the area postrema. And again, Martin has given you a little background on that. Area postrema GIPR neurons are inhibitory, and so this is very supportive of the fact that they are important given Randy's GABAergic knockout experiments. These neurons project locally within the area postrema, and the aggregate of the data so far has resulted in the following model, whereby in the area postrema we have GIP receptor expressing neurons that synapse onto the GLP-1 receptor neurons. And so agonism or activation of these neurons, which are inhibitory, would essentially affect to suppress nausea that's elicited by GLP-1 receptor agonists. On the other hand, if you antagonize the GIP receptor neurons, essentially silencing the inhibitory neuron that then serves to disinhibit the GLP-1 receptor neurons, you can imagine that you could increase nausea, but also suppress food intake. And so in this model, we have AP-GIPR receptor agonism that suppresses GLP-1 receptor-mediated nausea, and the antagonism will promote nausea but suppress food intake. But as I said, the cellular mechanisms that underlie the action of GIP on these neurons is unknown. So our questions, the questions we're trying to ask, is kind of to get at the this paradox, right? So how do GIP receptor agonists act on these neurons? And we can examine that directly with some of our methods, as I'll show you, and discover what are the cellular mechanisms underlying the action of these GIP agonists. Our overall goals are to gain some insight into the mechanistic action of GIP, both on these populations of neurons, but other neurons within the brain. And hopefully you can appreciate that the methods that we use can provide a framework for future testing of agonists, antagonists, coagonists, etc. Now, in order to directly examine the function of GIP on cells that express the receptor, we're using a mouse model that was developed by Frank Ryman at the University of Cambridge. And I won't belabor the specifics, but I would suffice to say that it's a GIP receptor Cree knock-in animal. And what this does, it allows us to both identify GIP receptor expressing neurons and manipulate those neurons selectively. And so shown in this figure here, in the AP, the top figure is just a staining for DAPI for all the neurons, and the bottom figure is a fluorescence indicator selectively expressed in these GIP receptor expressing neurons. Now, for our studies using this GIP receptor Cree animal, we can measure neuronal activity in a couple of different ways. One way that we measure neuronal activity is by measuring intracellular calcium or cytosolic calcium. Cytosolic calcium can be a surrogate of activity due to the fact that when neurons are activated, calcium can influx across the cell membrane, can also be mobilized by intracellular stores. And in order to measure intracellular calcium, we use a genetic model whereby we cross this GIP receptor Cree to another animal that has a Cree-dependent genetically encoded calcium indicator, GCAM. The thing to know about the GCAM molecule, shown in the top right, is that when calcium binds, you increase the fluorescence of this molecule. So essentially what we're measuring, an increase of fluorescence, which would be a surrogate of an increase of calcium inside the cell. To do this, we make acute brain slices that include the area postrema. We use both male and female mice. And these acute brain slices are kept in oxygenated artificial cerebrospinal fluid, so we can keep them alive for a few hours during the day. And as you can see in this video here, this is a measure of the GCAM fluorescence. And as it starts again, you can, well, yeah, there you go. You see some very beautiful spontaneous activity. At the very end, there's a huge increase in fluorescence activity, and that is actually an artificial stimulus we provide, high potassium, that we know will activate the neurons. And I want to point out that these studies, all the calcium imaging studies, were done by a very talented research specialist, Ainsley Caldwell, in our lab. And so when we measured the effect of GIP on intracellular calcium, in these area postrema neurons. What I'm showing here, and the traces on the left, are actually a normalized fluorescence signal. So we normalize the fluorescence to baseline. That's delta F over F0, and it's expressed here over time. And we see when we add GIP, 10 nanomolar in this case, there's a rapid increase in calcium in fluorescence, and then it diminishes in the continuous presence of the agonist. Also of note here is after we wash out the agonist, although I will say there's a caveat of washing out peptide from slices is a little bit tricky, so we're not sure the agonist is completely gone. But in any case, when that is washed out, there's a significant decrease in GCAMP fluorescence and neuronal activity. So we see this transient response in response to GIP application. There are also some cells where we see this very nice spontaneous activity, as you could appreciate in our little movie, of these calcium transients. When we look at the area under the curve to quantify these changes, the area under the curve is just going to be integrating the calcium response. We take the area under the curve over 2 minutes, and we look early on in GIP application and then later on. And as you can appreciate from the graph on the right, we see that there's a significant increase in the area under the curve early during GIP application. It diminishes over time, and then there's a significant decrease after we wash off the drug. When we look at this a different way, and this is a heat map from one slice, one example slice, so we can look at the population of neurons at the same time. You can see here, and each neuron is a different row. So we can see here that there's the nice transient activation in response to GIP. But we also found that there was a subpopulation of about 20% of our neurons that showed a more delayed sustained activation in response to GIP. So we see that there's these different subsets of neurons that are responding differentially to this application. And on the right here are just some traces that we see of these delayed sustained responders in response to GIP. So when we look at the area under the curve of these two functionally distinct groups, again, the graph on the right is just a recapitulation of when we looked at the total neuronal population. And the graph on the right is the looking at the end of the curve for the approximately 20% of neurons that display the sustained activation in response to GIP. So what we know so far is that we have a dual response to GIP application. This could represent two distinct populations. There is a transient activation in most of the neurons that diminishes over the time and then results in a suppression, a more long-lived suppression of activity. We just wanted to test that this activation was cell autonomous. We expect that it would be cell autonomous, meaning that the GIP is acting at GIP receptors on these cells that we're recording from. In order to do that, we bathe the cells in synaptic blockers. So we block action potentials flowing down cells. We block the detection of any neurotransmitter release so that we can just isolate these postsynaptic cell. And we find that, in fact, that the transient increase in GIP, that GIP-induced transient increase in calcium does persist with synaptic blockade. And you can see that here. Synaptic blockade, of course, causes a decrease in overall activity. We see an increase with GIP. We are still building up the power on this data, but so far we've not seen any neurons that show the sustained activation with GIP in the presence of synaptic blockade. So that suggests that that effect may be dependent from input from other GIP receptor neurons that may reside in the slice, for example, from the NTS. So we next directed our attention to trying to determine what are the cellular mechanisms underlying this effect that we've seen. And just to give you a very, very simplified overview of GIP receptor signaling, it is a GPCR, or G protein coupled receptor. And what that means is that a ligand, the GIP in this case, would bind to the receptor on the plasma membrane. And then at that point, this particular receptor undergoes a conformational transformation that triggers the binding of a certain flavor of G protein. This GIP receptor has primarily been shown to couple to what's known as the G protein, its excitatory pathway. And this pathway activates adenylate cyclase, which triggers the production of cyclic AMP. And in just a second, I'll show some downstream effects of that. The other G protein that the GIPR has been shown to interact with is a GQ G protein. This pathway is separate. And this pathway, for right now, I will just say results primarily in the result of intracellular calcium release. So since the GIPR is primarily coupled to the GS protein, and we know a little bit about the signaling pathways there, again, I'm just going to simplify this in a way that says that when binding of GIP activates the GS protein, we go through a series of intracellular signaling molecules, downstream of which is a kinase, protein kinase A, which can act directly on ion channels. So when these ion channels are modified, we can then increase ion flux across the membrane, change membrane potential, most likely with the GS coupling in a way that activates the neuron, will depolarize the neuron, and increase the firing of action potentials in this neuron. This also has the effect of, this membrane depolarization has the effect of increasing calcium influx into the cell through these voltage-gated ion channels. And so the hypothesis is that when GIP activates the GS signaling pathway, we get membrane depolarization, activation, opening of these voltage-gated calcium channels, and thus our rise in intracellular calcium. We can directly measure this using electrophysiology. And again, the way we do this is we make acute brain slices, we bathe them in our oxygenated solution, and then Jim Del Orco, who's done all of the EPHIS recordings that I'm going to show here, lowers a pipette with an electrode onto a single neuron, onto the membrane, seals onto the membrane, and if you apply pressure or suction onto that pipette, you can rupture that little bit of membrane. What this does is it provides electrical and chemical access to the cell, and then we can measure the ion flow coming into and out of the cell. What we're doing in this particular experiment is actually we're just sitting there and monitoring what the cell is doing. It's called current clamp mode. And Jim is measuring membrane potential and action potential firing. And what I'd like to get across with this slide is for most of our neurons, we don't see much of an effect, right? So this is not supportive of GS activation, modifying any kind of ion channel conductance. There are a few neurons, again, that show this more delayed, sustained activation, which may correspond to the calcium imaging that we saw. But in any case, it made us rethink the signaling, the intracellular signaling with the GIP receptor in the area postrema. I'm gonna skip over that. And what we started thinking about is maybe this is not calcium influx across the cell membrane, but maybe it's mobilization of intracellular stores. And as I said, the GIPR can also couple to a GQ pathway, mediated pathway. And in this pathway, with the activation of GQ, you activate phospholipase C, which again, activates a few intracellular signaling molecules, and the end result is mobilization of calcium from the endoplasmic reticulum. And so our hypothesis is maybe this intracellular, or this increase in cytosolic calcium that we're seeing is downstream of activation of GQ. And to test this, we applied a GQ inhibitor, or an inhibitor, a compound that prevents the binding of GQ, and that's YM254890. I'll call it YM. And in this experiment, we see nice spontaneous activity, calcium activity. We bathed the neurons with the YM compound, and to our surprise, they shut off. So just inhibition of GQ in the absence of GIP suppressed neuronal activity, or at least calcium dynamics in these cells. We then went on to add the GIP in the presence of the YM compound, and it had no effect. So blockade of GQ G protein signaling pathway prevented the activation of these neurons, or at least the mobilization of calcium in the presence of GIP. And just to test to make sure that our GIP is still working, we can still get a response to GIP, we did it the opposite direction. So we put on GIP first. We see here we have a nice response. Nice response to GIP. We wash the GIP off as we're washing the YM compound in. Again, we see a complete suppression of the calcium activity. And when we reapply the GIP, there is no effect. And again, this is shown in the heat maps here, and I think you can appreciate that these cells really go quiet when we put on this compound. That suggests that there may be some constitutive GQ signaling that's occurring, either from input from other cells in the slice or some constitutive receptor activity. So right now we're doing some more experiments where we're going to be following up on the role of this mobilization from intracellular stores and the GIP effect. And I'd be remiss if I didn't talk about another hypothesis in GIP receptor signaling, and that's to get to the question of whether or not there's receptor internalization and downregulation of the receptor. And although we haven't gotten to these experiments yet, we are aware that the GIP receptor, at least in adipocytes and in islet cells, can bind to a protein called beta-arrestin. And what beta-arrestin 2 does, then it mediates the internalization of the receptor, but it also is shown in adipocytes here, can slow the recycling of the receptor back to the membrane. And so some of the long-term suppression of activity that we see, or the decrease in the response that we see, could be resulting from beta-arrestin mediated internalization and sequestration of the receptor. So that is also an experiment that's on our list. There are some beta-arrestin inhibitors that we can check. So just in conclusion, I want to say that GIP, we have shown, induces two responses in area posterior neurons. One is an early transient increase followed by a decrease, and then there's a delayed sustained increase in calcium. Washout of the GIP resulted in a long-term suppression of activity. The sustained activation of these neurons may depend on synaptic input from other cells. And because we didn't see a very strong effect of GIP on our electrical activity, we do think this is due, the effect of GIP primarily is through mobilization of calcium stores, and it has a component of GQ receptor activation. So some of our ongoing and future studies, we want to examine the effect of GIP on other neurons, for example, in the NTS, and we currently have those data that we're analyzing. We have also been examining the effect of GIP on populations of GLP-1 receptor neurons, and our data support the fact that GIP receptors do inhibit GLP-1 receptor activation. We can also use this framework to look at the effect of other agonists and antagonists. And of course, it'd be nice to look at how the activity of these neurons differs under conditions of obesity or with long-term incretin-based therapies. And skip over that. And I just want to acknowledge former and current lab members, Ainsley Coswell and Jim Del Orca, who did these experiments, my lab manager, Alex, all my collaborators. Thank you.
Time for a couple of questions for Dr. Goforth.
Really interesting talk, Paula. I was wondering if you had looked at if the proportion of these transiently versus like sustained active neurons varies with body weight at all.
Oh, no, that's a good question. We've not looked at that. Yeah.
Really nice, Paula. Your cells that have the delayed sustained activation that looks like it's postsynaptic, do you think that's a change in excitatory input or inhibitory input, or I mean, what's your model for that?
If I were to guess, it'd be excitatory input. Mm-hmm. Yeah.
Coming from? It's not fair. Sorry.
Well, in the slice, the only other population that we have most likely is the NTS that have GIP receptor. Yeah.
Okay, other questions? Okay, thank you, Dr. Goforth.
Thank you.
Well, thanks again for a really tremendous presentation, Dan. And I'll ask the other panelists to come up and have a seat. And while they are, I do want to put in a plug for the University of Michigan here in terms of medication and other coverage. So all of you who are employees of the University of Michigan, I don't often have a chance to explain this, but I want to explain it for just a moment. So we're self-insured. Although it's administered by Blue Cross Blue Shield of Michigan, which is the predominant commercial payer, Blue Cross Blue Shield of Michigan made the decision not to cover obesity drugs, but the University of Michigan does cover those. So if I wanted to put a plug in for our benefits group at the university and the others who have gone into making this decision, because I think it's important. So now that everybody's up here, we have a great panel today, and I'll introduce them briefly. Mark Fendrick, who is a professor in medicine and general medicine. Carrie Ferrario, who's an associate professor in pharmacology. I didn't get a chance to see if you raised your hand. I'm assuming everybody's going to raise their hand. Dina Hafez Griauzde, who's a associate professor in internal medicine, and I apologize if I didn't get that quite right. Andrew Kraftson, who's an associate professor in internal medicine also. So what we're going to do, I have some questions, and I will direct them to one person on the panel who can provide an answer, and then for all the panel. And I think at each interval, if any of you have additional questions or would like additional clarification, please raise your hands and we'll do that. And we have till 12:30, so we have 35 minutes or so. So the first is to Dr. Kraftson. What are the side effects or downsides of incretin-based therapies?
Thank you for the question. So for those of you who think I'm not old enough to be on this panel, I actually remember prescribing exenatide in 2005 when it first came out for one of my patients that I was co-managing with endocrine. So I do have 20 years of experience of seeing how these medications have impacted my patients. And I think we're pretty familiar with the well-documented side effect profile. So gastrointestinal signs and symptoms are common. So I try to over-prepare my patients. I usually spend a good amount of time to say, hey, this is going to affect you and many people. So you have almost 50% of people who could potentially get nausea, depending on what studies you look at. And that is not a great fit with the Western way of eating food, which is scarf everything down, go about your day. And so we do talk about chewing very thoroughly, eating slowly, separating out eating and drinking, avoiding heavy fatty foods, and avoiding spicy foods for a little bit. And we talk about some analogies that you might feel like you ate too full and what would you do in those circumstances. So trying to normalize that this is a potentially a likely thing that could happen, you have to modify the way that you eat to help mitigate the danger of vomiting, because really we're saying that nausea may be a part of the process, but we don't want you to vomit. We don't want you to get dehydrated. We also know that approximately 20% of people will develop a change in their bowel habits, so people could get diarrhea or constipation. Many of the people that I treat have already had bariatric surgery, and so those are a bit more prone to constipation than diarrhea, but either is possible. And for some of these individuals, those bowel changes can persist for as long as they're on the medicine. It may not be temporary. The things that we really try to de-emphasize would be their concerns about pancreatitis or medullary thyroid cancer. As an endocrinologist, we're familiar with medullary thyroid cancer as a rare form of thyroid cancer, but really I try to go over in simplified terms that it seems that the C cells of the thyroid have receptors for GLP-1 that are not the same as for humans, and we're just not seeing it borne out over these 20 years of experience with the medicines. And same thing with pancreatitis. Certainly if you have obesity and diabetes, you're at higher risk of pancreatitis anyway, inflammation of the pancreas. But when we look back at the meta-analyses, the different cardiovascular data, the diabetes data, we are not seeing a lot of worrisome signals in terms of pancreatitis. So that's something that we can de-emphasize. But those gastrointestinal things are real. feeling crummy for a couple weeks. A number of people are reporting headaches, which is something that comes up in fatigue. And so that can be an issue as well. We do see the heart rate can go up about 10 beats per minute, but doesn't seem to be associated with malignant arrhythmias. Biliary issues can be at higher risk. You know, those can be a risk when you lose weight for any reason, but it seems to be slightly higher with use of these medications. And then there are things that are less commonly appreciated, which could be exacerbation of a pre-existing eating disorder if that wasn't already come up, or complete anorexia, and I use that as a neutral term, not really as just to say the total absence of hunger. So I can have patients who are doing very successfully with weight control, but then just like the other day, I had someone saying, well, I have 8 cookies and a yogurt, and that was yesterday's diet. And so that's where we're having weight success, but not true health success. And so we have to have a lot of shifting. So that's my long-winded answer to the side effect question.
That's great. Thank you. Other comments from the panelists?
I'll just say briefly that the biggest side effects is financial toxicity. And I know some of you are laughing, but in primary care for over 3 decades, it's pretty rare to see patients flocking into your office to have us do something for you. Usually we're trying to beg them to come in and reminding them the things that they're due. So I'm really grateful for Dan to bring up this issue and understanding that, you know, for us to really achieve the full population benefits of this amazing breakthrough, we not only have to deal with science and side effects, but also the delivery of these drugs effectively.
Can I ask a follow-up question on that
Sure.
topic? So you mentioned at the very end, right, you're making a small molecule drug, presumably, and what was salient to me was it doesn't require refrigeration, which presumably opens up a lot of other markets because not every place has the capacity that we have here. But is that going to be less cheaper to make basically and easier, or the capacity, will you be able to scale it up better?
That's the hope. That many of the things you just said are true. Of course, like any other manufacturing industry, scale is what matters the most. So right now we make a lot of tirzepatide. But if we can make orforglipron, if it's successful, we can make it at scale. It should be a more efficient process. It doesn't require sterile filling, which is, you know, the pictures of people in spacesuits working in a pharmaceutical factory. That's generally what they're working on. It doesn't. I think another benefit that I didn't mention is the environmental impact. Syringes are great, except for when they wash up on your beach. And so we need to make sure that future solutions have less environmental impact, and pills are much better.
And something I didn't mention before was that the need for aggressive de-escalation of other treatments. So they may need lowering of insulin or antihypertensives, and we're so used to go low and slow to build people up, but we may not be as prepared to de-escalate pretty aggressively.
Can I ask you a question in your experience? In some of the clinical trials, I've noticed some patients who are, with some of the newer medicines, losing weight so quickly that they're dissatisfied with the velocity of weight loss, that they feel like too many people are asking them about, are they okay, and their clothes don't fit. And is that something you're seeing in practice or not? Is it possible to lose weight too fast?
You know, yes, I would say it is. And one of the things that we're confronted with are various expectations, whether it's expectations from the insurer, whether it's expectations from the environment or the individual, that may not all align with what we want in terms of health goals. And so a slower titration process could be more tolerable, but some of our patients are under the gun, like, oh, we are going to track from the moment that you pick up the prescription, the clock starts ticking and you have to lose this amount of weight in this amount of time. And so then there's this pressure that, okay, I have this number, I have to achieve it. And that's not always in alignment with our health goals overall.
From the insurers as well, are you saying?
The insurers, yes.
Yeah, that's
So
kind of an interest. I mean, what other diseases do we treat that way in terms of your ability to control it with a That's very interesting to me that they would even have that cultural shift.
None, I'd say.
Yeah.
And what I said about the University of Michigan coverage is for 2 years. It's not forever. Other comments or thoughts from the panel? Well, let me ask you sort of a follow-up question. How many people don't respond to incretin-based therapies, and how might these drugs not work for some groups of people? That's for anyone.
I could start with clinical trial experience, but the real world could be quite different. In our clinical trials, almost every single patient had some level of weight loss, and I think upwards of 90% of patients had at least 5% body weight loss on the higher doses, maybe a bit higher than 90%. When I look at the waterfall plot of individual body weight, in every large enough trial, there might be one or two that didn't really have appreciable weight loss. I don't know if that represents a genetic difference or some other factor that made them true non-responders, or even if they just chose not to take the drug, which happens sometimes in clinical trials. So we're doing two things. Of course, it's simple, but I don't have that data, which is to get the PK and make sure that the drug was on board. And then if they're true non-responders, despite adequate exposure to drug, what we're doing across all of our patients, whole exome sequencing to understand can we find genetic markers, since clearly obesity is a heterogeneous disease. So not very many complete non-responders, but a large degree of variability, anywhere from 5 to 35% body weight loss. And we know some of the predictors, but not all of them.
I can kind of jump in on that too with a clinical perspective. So I practice obesity medicine at the Ann Arbor VA. And, I think in general, we sort of see an attenuated or diminished weight loss effectiveness of these medications in real world settings. I think some of it is related to patient level factors like perhaps the genetic variation that you're alluding to, or inability to tolerate side effects, treatment non-adherence. I think a bigger issue that we've paid considerably less attention to is sort of the system level factors that result in less weight loss than we would expect in clinical practice. So as Dr. Kraftson was saying, there's a lot of counseling that takes time. It takes time for patients to come back and have timely titration. What weight change have you had at month 1, and onwards until people reach the maximum dose. We have worked really hard to build clinical infrastructure that allows for weight-focused visits where that can be the primary topic of the visit. Patients are counseled. And then we have integrated pharmacists, nurses along the way during the titration period to make sure that patients are counseled adequately about side effects and that titrations are made in a timely fashion, because what you can imagine is in a general primary care practice, people are busy, they might get prescribed the drug, and then they may never be titrated, and then they lose insurance coverage. And so now you just have this cycle where you've wasted money, you have done nothing as a system to optimize patients' outcomes. And I think we really need to think more about how we deliver these medications. And Dr. Oshman will be talking about some of our work on that topic later today.
And sometimes we don't do enough with the needs assessment for our patients before we start them on pharmacotherapy to really see, have we adequately filled the health literacy and dietary literacy bucket? Have we really addressed mental health issues and emotion-provoked eating or other stress-provoked eating? And so that definitely will impact variability. But from the biology standpoint, I have patients who respond an A1C of 2% lowering. It's wonderful, but they haven't lost a single pound. And I have other people who have lost weight, but then they're still on insulin. I mean, just the variability is fascinating.
Well, thank you all. I'm going to move us to the next question, because I've got a lot of great questions here. So next is Dr. Fendrick. Given the cost and limited supply of current incretin-based therapies, what groups of people should be prioritized for treatment?
Thanks for that question, Dr. Runge.
An easy one.
No, as you know, I've been focusing on the access and affordability of really exciting but expensive interventions for over 3 decades here at Michigan. So as you know, in cardiology, the same issue arose with lytic therapy as it did in the early days of statins. And some of you in this room may not know that atorvastatin was actually developed at the North Campus Research Complex here in Ann Arbor. So we went through the idea that now that we're seeing it for pennies a day, we had to struggle to answer that exact same question, who would get it? So I think the issue of distribution is really important. And I'm so glad that Daniel brought this up, because if you look at most of the access issues for these breakthrough wonderful drugs, they're financially based. The decision to prescribe a drug for 2 years or till you reach a certain cap or that you have to pay an exorbitant amount out of pocket before you actually get coverage for it is particularly against the reason why many of us are in the issue of individual and population health. I think Dr. Griauzde will talk about the individual health issues, but when it comes to population, we want to get the maximum amount of health benefit for the money we spend. And for those of you who are willing to stay around for the closing talk, that's what I'll be addressing later on in the afternoon. But I will say that we have argued quite strongly that I didn't go to medical school to learn how to save people money, nor did I go to medical school to learn how to make people money. If you go back and read my personal statement, it was like, a bird fell out of the nest and it broke its wing. I fixed it, it flew off, and I wanted to be a doctor. So, so that's what it was. And we have seen this unmet need for my entire career. The fact that we have these agents, and not just these drugs that we talked about, multiple ways to help people effectively lose weight, and a majority of people in the US can't get them, suggests there's a problem. So how do we do it? We have real clinical experts who also know a little bit about economics and finances to figure out where can we put the money to get the biggest bang for the buck. For statins, it was secondary prevention in the early days. We couldn't give it for primary prevention. We had the same step therapy and other types of things going on. So we're living this issue all over again. My point, given the effectiveness on these agents, and even further validated by Daniel's spectacular review, is we need to expand coverage of these drugs. There is absolutely no question. And while all the clinicians and researchers are saying, yeah, that's what we want, there's someone in the back says, there's Fendrick again spending my money. The Congressional Budget Office estimated that $35 billion would be spent if we covered these things for Medicare. And some of you might have seen just last week, the Trump administration decided not to cover these drugs. And it's very, very frustrating to me to think that we have the money to pay for these, in my opinion. The first thing we need to do and understand, statins are a good example, hepatitis C medications, a good example. Prices will fall, right? I pay nothing for my statin, thank you, Marshall. And I think you as an employer pay very, very little for that statin right now as the ultimate purchaser. So prices will fall, give us some time in that regard. We don't need to be on the drug forever. Daniel and I may have a disagreement on that. There are de-escalation ideas underway that maybe we limit not the prescription to these drugs based on time or money, but we actually have clinicians say, this is when it works best for you. And there may be other alternatives that Dina and Andrew would come up with that say may be as good, but less expensive down the road. And the last thing, which is really important, and I'm working with Daniel's colleagues at Lilly around this, is that employers and payers are spending billions of dollars on things that we wouldn't buy even if they were free, right? So why argue with me about something that's great that's too expensive, like these drugs are my iPhone, while employers around the country are still spending billions of dollars on blockbuster memberships, right? So we have to instead of arguing about good stuff being too expensive, really focus on the billions of dollars that I think I could work with Daniel and his team about to cover the entire cost of these drugs for Medicare.
Thank you, Mark. Other panelists like to comment on that? Very well said. Go ahead, Dina.
Oh, I was just going to say, I think that there are other weight loss treatment options that can be highly effective and have been severely underused. So if we look at our older anti-obesity medications, we sort of created this narrative that we've never had a treatment that can support 20% weight loss. That is true on average, but all of these treatments have wide individual level variation. Weight loss medications, bariatric surgery, lifestyle change programs. There are a lot of patients that do significantly better than or worse than the average. And when we've only used other effective treatments in 2% or less of the population, we have not realized the individual patient benefits of cheaper, older medications, and we certainly have not realized their population health benefits. So as an example, I said I practice clinically at the VA. We've developed a lot of population health management tools so we can look at all of our patients in aggregate. When I look at my patients in ranked order of who has lost 20% of their body weight or more, 1 is on tirzepatide, 2 are on semaglutide, 1 has had bariatric surgery, and I think the remaining 5 are on Qsymia, which is phentermine topiramate, or bupropion naltrexone. So it is false, it is not true that we can't get to 20% weight loss with other treatments. And we, again, kind of going back to what I said in my first point, we really need to think about the right system of care delivery, because the cost question is not how do we get every patient with obesity an incretin-based therapy. The cost question is how do we use these medications in a judicious way, a patient-centered way. And last point, kind of a tangent, but if we are using these medications in younger patient populations, we have to acknowledge we don't know their long-term side effects. That's just a reality. We don't know. We have to be humble enough to acknowledge uncertainty and give our patients the opportunity to lose weight with medications that can be effective and potentially safer.
Thank you.
Yes. Add one small piece to that, which just because it might not be familiar, particularly maybe to some trainees in the audience, they do bariatric surgery on children. And so they are being treated with these other interventions, which are some forms non-reversible, some newer forms actually reversible. But so I think, when we talk about kids, and that's a really important point, right? These are long-term biological changes, both with obesity, with type 2 diabetes, and now with whatever drug treatment or surgical intervention you choose, right? And so in the case of children, we're always more cautious, as we should be, and particularly through development and reproductive health. But they are doing surgeries on children now because the outcomes for liver failure are so dire, really, as I understand it. So I think it is really important for us as a research community to think about that as well on the basic science side as well, and the models we have and what we can learn about what might be safe, because drugs are great, you can stop taking them, and the body can recover, and it's incredibly plastic. So I thank you for bringing up those points.
Thank you all. I want to make a plea here for the panel and for all of you. We have a lot of smart people here. I think a fundamental issue in healthcare in the United States is that in many instances it's looked at the costs on a quarterly basis or an annual basis. So whether it's a medical insurance company or whether it's the University of Michigan, we're coming up to the time of year where we have to get our budget approved by the regents every year, that this is a reality on the one hand. On the other hand, there have been studies ever since I've been in medicine about the cost per quality, per year of quality life expectancy. And I don't think the incretins have been around long enough to do that, but I think a really important thing that I'd ask you all to think about, and I'm not the person who can do any of this, but to think about how do we promote consideration of therapies, incretins and others, that will have a very profound impact on both quality life expectancy as well as financial issues for the United States going forward and the world. So that's just a brief testimonial, and I'll move on. The next question is for Dr. Ferrario. It says, many insurance plans will only cover incretin-based therapies for 1 to 2 years, at least for the obesity indication. I wasn't trying to jump in on this question, but how can we justify treating people for this amount of time, given that most will eventually regain the weight that they lost? And should we be pressuring insurance companies to provide lifelong coverage for incretin-based therapies?
So I am a basic sciences researcher, so I study rats mostly. And my background is actually in motivation and addiction and substance use disorder. And I came into this field when I started my own research group about a little over 10 years ago now. And so the color of that on me is again, just thinking about health and quality of life. And it's kind of crazy that we say, oh, well, there's a limit on this. And I'm thinking about how we treat people with substance use disorder. You get 6 to 8 weeks in a treatment program, and then you're supposed to be fine, and we know that doesn't work, right? Because it's a disease, it's a brain disease, and obesity, diabetes, these are all biological diseases. So it is interesting that because it's not perfect, this is, I think, more psychological and cultural than anything. Because it's not perfect, it's not good. And so one of the things that we think about, the term we use in other areas is harm reduction, right? So any period of time with healthy weight, with healthy A1C, that's going to reduce harm. And we know, for example, that you have compromised blood flow, you're going to have increased inflammation, you're going to get all of these knock-on effects, like Daniel was mentioning about treating potentially cognitive decline, right? So I think any period of time where there's health benefits that we can really operationalize is good. And really, how long can you stay on the drugs in a safe way, I think, is the question, more so than, so what if you regain the weight? Okay, fine, right? So what? I mean, I think that kind of goes back to that mindset of somehow it's a moral failing or somehow it's your fault, right? And we have to remember that we're calling a lot of this success when people aren't changing their dietary habits. They're not necessarily eating more healthily, as my colleague pointed out, cookies and yeah, you can go on the Twinkie diet, right, and lose a bit of weight and not be healthy and have a whole other bunch of other knock-on diseases, right? So I think that's more of a cultural piece in a way, and then of course the monetary side of things, which we just kind of can't escape in the U.S. But I am curious what the others think from their clinical perspectives.
So Marcia, I'll chime in. I'm going to behave a fee for service as opposed to capitated, because this question is right for me. So on your tangent, we as a society and the National Academy is struggling with the issue of these multi-million-dollar curative therapies, many of which are being studied by researchers here at U of M. So the idea of laying out millions of dollars to prevent some of these conditions, whether it be through gene therapy or others, is just remarkable, and that's why we became clinicians. But the reality, your point about this idea of budget impact, when I was told by one of the big three automakers in 1998 that I had a quarter to show my return on investment, I thought that was 25 years and not 3 months. So we have to realize the constraints that these are under, and many of the researchers in the room know that the law of the land actuary, the Congressional Budget Office, is limited to 10 years by law. So as much as you and I want to look at longer, they'll tell you what the bill is, and that $35 billion number is what's out there. There is an element in the Washington Post from last week saying that if we invested, we'd actually save money, which I don't agree. But I think we would have significant offsets, some of which we measure and some of which we don't. So there are people looking at this now, including some of Daniel's colleagues at Lilly. But the idea of having a non-clinical decision of when you stop and start is something that you can take a lead on. Like the 2-year idea, if I were to ask these experts here, is like, how many didn't need 2 full years and how many people need more? And you get the actuary to come in and say, oh, maybe it could be some type of clinically driven approach. There's all this pushback, as you know, about prior authorization and utilization management, but it's never on the patient's behalf, right? I think that we've thought long about in this area and others that we take advantage of the clinical expertise and let the benefit design follow as opposed to the other way around.
Thanks, Mark. I want to be sure we get to all the questions. So next is for Dr. Griauzde, which is, are there any useful non-incretin-based weight loss therapeutics available? And if so, when should they be used? You commented briefly on that, but I know we'd all like to hear more about that.
Yeah, sure. Yes, I guess I did sort of answer that in my first response. So yes, there are definitely pharmacologic and non-pharmacologic effective alternatives. We can consider lifestyle change programs, more intensive medical nutrition therapy interventions like meal replacement programs. I personally do a lot of low and very low carb dietary interventions. And then of course, older anti-obesity medications, bariatric surgery, endoscopic bariatric therapy. All of these things can work. I think I acknowledged in my first statement that we focus on average treatment effects, but really there's wide individual level variation and outcomes with all of these treatments. And our greatest challenge is helping patients navigate the full range of treatment options, taking into consideration, individual level preferences. At this point in time, not everybody wants to be on a weight loss medication at all or an injectable weight loss medication. Certainly not every patient wants bariatric surgery. So we need to sort of have the time to explore patients' preferences and then tailor the treatment based on individuals' preferred treatment path. Unfortunately, at this point in time, we don't have a lot of sort of reliable pre-treatment predictors of who's going to do well with any treatment. In an ideal world, Patient would come in, there would be some laboratory studies or intake form that could help guide you. And we don't have great pre-treatment predictors at this point. One thing that is fairly reliable is this idea of early weight loss response, and that we see across a variety of weight loss treatment options. People that lose weight within the first 3 months tend to do better longer term. And so it's super important to help your patient pick a preference-sensitive starting point and then have the clinical infrastructure in place so people don't fall through the cracks. You can see people back at 3 months or have another strategy in place to touch base and then iterate, right? People have struggled with obesity for long periods of time, and so it really is not, I think there's no harm to be done to try the patient's preferred starting point as long as you have a check-in and potential for future treatment options.
Okay, can I ask a follow-up question?
Yeah.
So we're working on lots of different mechanisms and new drugs for the coming decades for obesity, but what attributes would you like to see us engineer into medicines? Is it more efficacy or better safety or once-a-year treatment or cheaper? What's the most important thing if you pick one?
Oh, geez.
Or better predictability, which is what you're talking about.
Cheap would be nice. Less expensive. Yes,
yeah,
less
okay,
expensive,
that'll happen
effective, less
naturally, yeah.
expensive.
These drugs will come
more
off patent.
affordable.
They'll be free.
Yeah, I don't know how to answer that question, just picking one. I think all of those are really important, and I think there's just a lot that we still need to learn. And maybe, to your earlier point about, who are these non-responders? If they're taking the drug but not losing weight, why is that the case? I think certainly in clinical practice, we see that patients, some patients, I had 2 patients last week coming in on tirzepatide that hadn't lost any weight, and their experience is that nothing has changed for them. They feel exactly the same. So sort of understanding more about that, I think, is very important so we can tailor treatments.
Thank you.
Yes.
I think that this gets to a question that I think is on your list, is sort of how we can think about what we're treating. So like you want to know who's going to be a responder, right? And then asking, I think it's interesting to ask, what is the root cause? Some people, it is just poor diet and exercise and their basal metabolism, right? That's just sort of where they're at, and they've tried, and it's going to work for them. But other people, there might be comorbidities with some underlying eating disorder. There might be comorbidities with depression and anxiety, which is something that I know we wanted to talk about a little bit. And so I sort of wondered to what extent we can use some of the patient data to inform perhaps what they should be modeling and asking about, or what we should be modeling and asking about. Because the question is if the root cause is A versus B in terms of what's causing the weight gain initially, right? Does that matter? And my hypothesis is I bet it matters. I bet it matters a lot in terms of treatment outcome, patient compliance, which one of these approaches, pharmacological or not, is going to work for your patients. But how do we build that into the types of studies and the capacity and the motivation that Lilly clearly has, right? I mean, it's great. They're going to come up with a lot of other approaches, but how do we build that into our rodent models and preclinical models? How do we use the patient data we have? And I don't know if you collect, you guys collect that type of information as you're going, or you have some inferences of what patients you can see it works better for or not.
Well, it's interesting. We can ask about satiation, satiety. Yeah. We can ask about cravings. But, food noise was not a term that we really used 10 years ago. And to hear people describe something so vividly has to, in my mind, lead to some underlying mechanism, because other people, they don't have the food noise and they didn't have any improvement once they've been on treatment. So I think those things are fascinating.
Yeah, that's one of my favorite phenomena. I think that like the idea that, you just sort of know that the pasta is in the fridge and it's just sort of there in the back of your mind. So this is what I find fascinating about the brain, are these motivations and these cravings and what you know is there and what you could be doing instead of what you're doing right now, which might be eating, right? And a lot of people do report that chatter just kind of goes to the background. And that doesn't seem like the same thing as satiety, right? And fullness. These all seem very, very different. And those are areas that I think a lot of us here are really interested in or studying. And so those concepts, I think at the basic level and at the human level, bringing those into the types of R&D that's being done in the pharma spaces would be really useful.
Yeah, these are great points. And we only have about 5 minutes left, so I wanted to ask Dan one last question. And that is, you commented a little bit about what I'll put into the big category of addictive behaviors. And I find that just totally fascinating because I have also heard the same thing about people either stopping smoking, who've been trying to stop smoking forever, or having a decreased urge for alcohol, not an aversion to it, as you said. So trying to look ahead when the studies haven't been completed, what do you think about the potential for incretin therapy in these? What is it the same mechanism that's affecting all these people, or is it different mechanisms? Is it the
I'll start, but then others may help out. Maybe the easiest answer is like, we don't really know yet, but we will find out. And I think, Carrie, you were talking about the food noise thing, which it's kind of interesting because when we finished the phase 3 clinical trials, I asked my team, so how does it work? Like, what's the patient experience? And they're like, oh, we're not really sure. And as soon as we launched it, like all we heard back from patients and doctors, and like you could read it on social media, is the food noise has gone away. Now this must be what everyone else feels like, people who don't suffer obesity. And so like this experience of taking medicine is different than the things we measure in clinical trials, which is interesting. And the addiction stuff mostly comes from reports of patient experience. There's now some real world evidence and retrospective looks, and maybe some very early randomized trials. But the patient experience is almost exactly the same words, that like they're thinking about addictive behaviors less, and they'll talk about drinking and smoking, and they'll also talk about shopping or gambling if they have those issues, which is interesting. And so why would those things be linked? Now I'm way off in hand-waving speculative territory, but you could just imagine that we've evolved to be like really good at seeking food and being rewarded for eating and storing energy. That's a good survival instinct. And some of these other behaviors that are addictive hijack some of that same brain circuitry, and maybe these drugs could help, but we need the data to find out. Maybe you have more.
Yeah. I mean, that is the foundation of substance use research, is that's what these drugs do. They hijack systems that were essential for our survival, that normally drive behavior towards reproduction, food, sex, shelter, and safety. Should I run away? Should I fight? And that, a lot of these players, so leptin receptors, are located on dopamine neurons. Dopamine systems are really important for shaping motivation. Do I move towards something or run away from it, right? So the pieces are there. What's really interesting to me is, drugs of abuse jump over all of this really beautiful biology. They just act directly on dopamine systems, whereas one thing that struck me about the data you were talking about at the end is now we're getting to multiple components of these integrins, right? Food is activating this really coordinated whole body physiology in the way that it evolved to be activated. And so I think that difference is really important, particularly for thinking about treating substance use disorders with these types of medications. There's really beautiful work coming out of Penn. So Sue Grigson and Matt Hayes and Heath Schmidt and Ziva Cooper are starting to ask how these drugs affect people with substance use disorder who are treatment seeking. And the one point I want to make, last thing, is that alcohol, I think, is one of the more interesting ones. There's really good data from NIDDK researchers about bariatric surgery and how it can shift alcohol, and mechanistic studies there talking about what's really shifted. Alcohol has calories, and you usually eat it with other calories, right? Beer is like bread. But I think that's really important, because that's the one where I think there's the best mechanistic evidence for these drugs working. Whereas, thinking about the extreme, the psychostimulants, amphetamines increase metabolism. Amphetamines are great weight loss drugs. Every time they find a weight loss drug, like in the past, turns out it was an amphetamine and it was highly addictive. So we don't take those. People might remember fen, for example. So I think that there's a lot of excitement, and there could really be something important there. But just like the etiology of obesity matters, and with addiction, I think it's complex and it's going to matter. And the patient population really matters. Taking some of these drugs is not going to be safe for people with liver cirrhosis. It really depends on where they are in the progression of their disease. So.
probably the outcome we measure matters too.
Oh, of course, yeah.
I was struck by your comments
Yeah,
on
weight
harm.
loss is not the measure. And it's going to cause weight loss, which is not good for a lot of people.
Well, but your comments on harm reduction, right now, I think, the FDA requires abstinence as the outcome for alcohol use disorder studies, which is probably not something to expect from this class of drugs.
Yeah, which is something that researchers in that space, clinical and preclinical, have been arguing for a really long time, is that, harm reduction is a benefit. It does help. It prolongs life. It prolongs health. It enables people to work, because we always like to monetize suffering. And so it enables people to work if they can function, right? And that also makes them feel better, and that helps them to really engage. So so I think the outcomes really matter. And for this, you'd have to ask, what is the outcome? And can you convince them that complete abstinence is just a kind of a ridiculous bar? And I think we all know that in the research space, but we just kind of haven't been able to push that through in other levels.
We'll do our best. Data is what matters. So once we get the data, we'll work on it.
Well, I'm looking at the clock. It says 0, 0, 0. So I just want to thank all of you for coming today. I hope that this morning has been as stimulating intellectually to you as it has to me. The message I'm taking home is the incretin system has lots to learn about it, and I'm so glad we have leading investigators working on that. And I want to thank the panel. This was a fabulous conversation, and Dan for spending his time with us and helping stimulate all this conversation with the work that you've led at Eli Lilly.
Thank you. My pleasure. More to come this afternoon.
Yep.
Okay, well, I'm going to get started on the afternoon session. Okay, welcome back to the afternoon session and the lightning portion of the symposium. So we have 5 poster presenters who were selected by faculty review to give 5-minute talks. Okay, so each of the lightning presenters will be given a $175 gift card. Now you're probably wondering, $175, where's that come from? Okay. So this is in honor of our Dodransen bicentennial. What does that mean? And everyone got quiet. And they're quickly looking on their cell phone saying, okay, what is this? How do I even spell that? Okay, from what I understand, it means a quarter century less than a bicentennial. So if you do the math, 175. Ta-da. Okay, so you get that. So when you're done, I'll give you your little envelope. Okay. So we got everyone back to the seats. This is great. I did my job. So there will be no questions for the speakers. They're going to do an awesome job. They're all super psyched to do it, super stressed. So let's show them some love. Okay, reduce their stress. But if you do have questions, we have beer and wine, but apparently no tequila. Sorry. Okay, so that's during the reception which follows. Okay, so let's please enjoy it. So our first speaker is Amanda Kucinkas, my apologies. She is a Ph.D. candidate in the Department of Molecular Integrative Biology or Physiology and laboratories of Aaron Giles and Durga Singer. Her title is Impact of Incretin Mimetic Drug Tirzepatide on Obesity-Associated Breast Cancer Growth, Findings from a Preclinical Model. Thank you.
Okay, hi everyone. I first wanted to just thank the organizers for the opportunity to speak today. I'm really thrilled to kick off the lightning talks and also excited to hear from the other lightning talks that come after me. As Dr. Jepson said, my name is Amanda Kucinkas, and I'm a second-year PhD candidate in the Department of Molecular and Integrative Physiology. And I will be speaking about an ongoing project in the Giles Lab titled The Impact of the Incretin Mimetic Drug Tirzepatide on Obesity-Associated Breast Cancer Growth. As I think everyone is aware of in this room, obesity is a very critical public health concern. And in the Giles Lab, we're specifically interested in obesity in the context of women's health and breast cancer outcomes. Obesity not only increases the risk of developing breast cancer, but it also contributes to poorer outcomes, including increased risk of developing metastases, drug resistance, and overall mortality. And while weight loss attenuates these cancer risks, these groundbreaking incretin mimetic drugs have sparked significant interest in the potential use for cancer prevention. I forgot that I had a slide. Here I am highlighting two preprints from the labs of Dr. Michael Polak and Dr. Steve Hursting, demonstrating that tirzepatide, which as we've talked extensively about today, is a dual GLP-1 and GIP receptor agonist. And this drug significantly reduces the growth of both colorectal and hormone-independent or triple-negative breast cancer growth in a diet-induced obese mouse model. Though there has not yet been investigation of these incretin mimetic agents on hormone-responsive breast cancer subtypes, which account for approximately 80% of breast cancer cases annually. Thus, this was the objective of our current study. So just briefly, we placed 9-week-old female C57 black 6 mice on a 46% high-fat diet to induce obesity. At 32 weeks of age, they were randomized to every other day injections of either tirzepatide or vehicle control. And 3 weeks later, or at 35 weeks of age, ER-positive tumors were initiated by injection of PY230 cells. Over the course of this 16-week intervention, we took weekly measurements of key metabolic parameters, as well as monitored tumor growth. The main finding of this study was that tirzepatide reduced the size of ER-positive mammary tumors in this diet-induced obese mouse model. In terms of metabolic parameters, the tirzepatide group demonstrated a 20% reduction in body weight, and this was primarily due to a reduction in adipose mass, which also averaged at approximately 20% and was sustained throughout the intervention. When the adipose mass was further divided into depots, we saw a significant reduction with tirzepatide on both visceral and subcutaneous adipose depots. Here, I'm highlighting the visceral adipose depot as it's a key marker of metabolic health. Lastly, what I'll be showing today was that with the tirzepatide group, we saw a significant reduction in liver fat, which, as you can see on the left, was quantified by qMRI, and some representative H&E images of vehicle versus tirzepatide livers on the right. Interestingly, upon correlating these metabolic markers with tumor volume at the end of the study, we found strong correlations between tumor volume and all three of these metabolic parameters, including body fat here on the left, maybe, visceral adipose here in the center, and percent liver fat on the right. And we are tangentially working to elucidate these mechanisms that are linking obesity and ER-positive breast cancer. If you're interested in hearing more about how we are continuing this work in postmenopausal animal models, investigating direct effects on hormone-responsive breast cancer cells, and incorporating exercise to prevent lean muscle loss, which we discussed earlier, please come see me during the reception at poster number 18. Thank you.
Our next speaker is Jen Volosik, also a PhD candidate in the Department of Molecular Integrative Physiology in the laboratory of Alison Affinati. The title of her talk is Stressed Out, Glucose and GLIPR1 expressing neurons as keys to uncovering the neurocircuitry that controls stress-induced hyperglycemia.
Alrighty, let me just make sure. Ooh, Amanda, I see your issue. Okay. Well, thank you for that introduction, and thank you to everyone who's been working so hard behind the scenes to make today so great so far. And finally, thank you to the organizers for inviting me today to give a brief overview of my dissertation project, which focuses on uncovering the neurocircuitry behind a clinically relevant problem of stress-induced hyperglycemia. So to begin, I want to talk a little bit about stress. So individuals can experience many distinct types of stressors. There can be physical and physiological stressors like tearing a muscle or getting a bacterial infection. There can be broken bones or heart attacks. Additionally, there can be psychological stressors, such as giving a lightning talk or just generally being a scientist in 2025. And although these stressors seem quite distinct, there are several physiological processes that are conserved throughout them in response, one of which being an increased metabolic demand of peripheral and central tissues. In order to meet that increased metabolic demand, the body must provide fuel, and that fuel source is going to be glucose. So under all of these stressors, there's an increase in blood glucose, and this is where the term stress-induced hyperglycemia comes from. To get into the mechanism of the very important process of stress-induced hyperglycemia, it occurs in two overlapping phases. The first phase, or the rapid phase, is mediated by sympathetic nervous system, or SNS innervation, of peripheral metabolic tissues, primarily the liver. This allows for the increase in gluconeogenesis and suppression of glycogen synthesis. Additionally, there's the sympathoadrenal response. This allows for the SNS to communicate to the adrenal medulla and allow for the release of catecholamines. The second, or sustained phase, involves the hypothalamic pituitary adrenal axis, or HPA axis mediated release of cortisol, our stress hormone. There's also central and peripheral mediated release of cytokines like interleukin-6, 10, and TNF-alpha. Taken together, these two phases and four components allow for the immediate and then sustained rise in blood glucose in order for us to adapt to the stressor at hand. And although this is a very important survival mechanism, we see in clinic that there are many cases of what's called dysfunctional SIH, including more than 50% of ICU patients experience excessive SIH, which is correlated with an increased risk in infection, increased risk of developing metabolic syndromes, and increased risk of hospital mortality. On the other side of that, inadequate SIH is also associated with an increased risk of mortality. This demonstrates that the system needs to be tightly regulated in order to remain effective. Treatments and efforts to maintain this tight glycemic range are mostly glucose and insulin intravenous infusions. However, this treatment has also been associated with an increased risk in mortality. Therefore, there's a clear critical clinical need to understand the mechanisms underlying stress-induced hyperglycemia so we can then better treat patients in the future. To take a basic sciences approach at this problem, we in the Affinati lab study the center of this mechanism, which is the brain. Within the brain, there's a region called the ventral medial nucleus of the hypothalamus, or the VMH. The VMH is very important in behavioral and physiological responses to stress. More specifically, in the dorsal medial and central component of the VMH shown here, there's subpopulations of neurons that are very important in mounting an SIH response into many different stressors. In rodents, we use psychological stressors such as restraint or hypoglycemia. So in order to dissect this central and dorsal medial component further, we performed bioinformatic analysis and discovered GLP-1 expressing neurons. And I'm so sorry, but for the first time today, I do not mean GLP-1R. GLP-1s, but they're pretty cool as well, I promise you. When we did functional analysis of these neurons, we found that they are necessary for restraint-induced hyperglycemia, which again is a psychological stressor, but not for insulin-induced hypoglycemia. Upon further looking into the mechanism of how this attenuated blood glucose mobilization may be occurring, we found that this could be due to an attenuation in the secretion of corticosterone. Therefore, we believe that VMH GLP-1 neurons represent a highly specialized neurocircuitry that controls SIH under psychological stress, but not other types of stressors like hypoglycemia. Future directions will look into other types of stressors such as inflammation with LPS and other mechanisms of how we may be controlling the HPA axis. Ultimately, these novel findings allow us to better understand the SIH neurocircuitry and will guide future therapeutic developments for dysfunctional SIH in clinic. That is all.
Okay, next up is Marina Blayler. She's a postdoctoral research fellow in the Department of Molecular Integrative Physiology, Laboratory of Lei Yin, and title of her presentation is Uncovering the Role of Hepatic FBW7 in Alcohol-Associated Liver Disease. Marina?
Thank you for the nice introduction, and thank you to everybody for your interest, and I would like to thank the committee for giving me such a great opportunity. Okay. Okay. Alcohol-associated liver disease, or ALD, is a progressive metabolic disease that starts with accumulation of fat in the liver, namely steatosis. Actually, you can see, okay, you can see right here. But some patients can advance to steatohepatitis, which is characterized by liver inflammation, fibrosis, and injury, and further advance to liver cancer or cirrhosis, which can result in liver failure. And the only treatment option for this condition is a liver transplant. The prevalence and the mortality rate of ALD is reflected by the fact that in 2023, 44% of liver disease deaths in the US were ALD related. Still, there are no targeted therapies for ALD. Its precise mechanisms are obscure, and research in this field is urgently needed. Disrupted processing of lipids is a hallmark of ALD. Thus, we focused on the role of the key regulator of lipid metabolism, FBW7, also known as FBXW7. It is a component of an E3 ubiquitin ligase complex involved in degradation of various hepatic factors. Hepatic loss of FBW7 was in mice on non-alcoholic diets was recently shown to promote steatohepatitis by inhibiting the activity of the nuclear receptor PPR alpha, a critical regulator of fatty acid oxidation. We questioned whether hepatic FBW7 plays a protective role in the ALD mouse model, and whether it exerts its protective function through PPR alpha. And if so, could PPR alpha agonists mitigate the effects of FBW7 loss? To answer the questions, we conducted experiments in mice where with flox FBW7 allele. We deleted FBW7 through injection of the adeno-associated virus carrying CRE recombinase on the hepatocyte-specific promoter. And these mice were fed alcohol diet, ethanol diet for 10 days, plus a single binge at the end to promote liver injury, acute on chronic liver injury. This approach was developed by Dr. Gao and coworkers to mimic human patterns of alcohol consumption. Our results showed that hepatic loss of FBW7 reduced expression of PPR alpha and dependent genes and suppressed fatty acid oxidation pathway in the ALD mouse model. Consequently, these mice were more sensitive to the alcohol diet-induced inflammation and steatosis and liver injury. This is shown in red, FBW7 knockout. Furthermore, an addition of PPR alpha agonist fenofibrate reduced liver injury, which is shown in blue, and reversed fatty liver. You can see here. Okay. Bulk RNA-seq analysis showed that FBW7 knockout was associated with enrichment of the differentially expressed genes in the thyroid hormone pathway, which is some of those are shown on this heat map. Notably, SERPINase7, or TBG, which is known as a major transporter of the thyroid hormone in circulation, and which is produced by the liver, was highly upregulated in FBW7 knockout livers, which is likely due to downregulation, overall downregulation of the thyroid hormone pathway. The qPCR data showed that expression of SERPINase7 is both dependent on both FBW7 and PPR alpha. And this is an exciting finding because it is known that thyroid hormone and PPR alpha pathways are cross-regulated. So in the future, we will focus on the FBW7 PPR alpha thyroid hormone signaling axis. And we hope that this will help to determine potential targets for treatment of ALD. With this, I would like to thank my primary advisor, Dr. Lin and Dr. Xin Tong, my lab members, my T32 training program in basic diabetes research and its leaders, Dr. Armand McDougall and Dr. Peter Arwin, the National Institute of Health, the National Institute of Diabetes and Kidney Disease, and thank you for your attention.
Okay, the next presentation will be by Dr. Jin Lee, a postdoctoral research fellow in the Department of Internal Medicine, Division of Metabolism, Endocrinology, and Diabetes, in the laboratory of Scott Solomanpour. Title of her talk is LAN-P1 Regulation of Mitochondrial Protein Folding Provides Insight into Beta Cell Failure in Type 2 Diabetes. Dr. Lee?
Thanks for your nice introduction. Good afternoon, everyone. My name is Jeannie, a postdoc fellow from Scott Solomanpour Group. And today I'm super happy to share my postdoc project on the LAN-P1. And this work was just accepted, officially accepted by Nature Metabolism today morning. So. As we know, pancreatic beta cells are the primary source of the insulin in our human body. And for the patient with type 2 diabetes, unfortunately, they don't have sufficient amount of the insulin and to maintain their normal blood glucose levels. And due to the loss of functional pancreatic beta cells and insulin resistance. And emerging evidence also indicated that type 2 diabetes is a protein misfolding disease. Two well-studied examples include misfolded proinsulin and also aggregated alert amyloid polypeptide. And these misfolded proteins are toxic to the beta cell, and which lead to beta cell failure and eventually contribute to the development of type 2 diabetes. And however, for the general picture of protein misfolding in type 2 diabetes, such as the extent, location, and the specific impact of the protein misfolding in type 2 diabetes is unclear. And in order to address these questions, firstly, we performed the quantitative proteomics and on human alert from the donors with or without the type 2 diabetes, and to look at the protein solubility on these patients. And when we received the human alert, this human alert was separated and into soluble and insoluble fractions with a buffer containing 100% and Trita X100. And for the insoluble fractions today, I would like to focus on because these fractions contain misfolded proteins or protein aggregates. And from these volcano plots, we also can see, and in type 2 diabetes, and this alert, and if we look at and carefully, a large proportion of the proteins, they are differentially enriched in the insoluble fraction of type 2 diabetic alert compared to the human alert from the control donors, which is super interesting. And then we first look at where they are from, and the cellular component based on the gene ontology analysis. And interestingly, we observed some misfolded proteins there from the ER, which is quite interesting for us because ER stress or ER protein misfolding, they are often considered as the major contributor to type 2 diabetes and to beta cell failure. And most surprisingly for us, we also observed a large number of these misfolded proteins. They are also from the mitochondria, and especially from the mitochondrial matrix, and which is also quite interesting for us. If we look carefully, we also observed the LAN-P1 protein, which is a very important mitochondrial matrix protease. They are also significantly enriched in the insoluble fraction of type 2 diabetes. And then based on these observations, we can say mitochondrial protein quality control may be impaired in type 2 diabetes. And the next, we utilize the single-arm sequencing data and to look at the expression of all mitochondrial matrix chaperones and protease. And interesting, again, we observed the LAMP1 and in the type 2 diabetic alert was significantly and downregulated compared to the control donors of this alert, and which is quite interesting. And we also performed the Western blot to look at the LAMP1 expression, protein expression, and we also can say LAMP1 protein expression was also much lower and in the type 2 diabetic alert. And with this piece of data, I got new questions again. And how long P1 and regulated beta cell survival and proper mitochondrial protein folding and in type 2 diabetes. And if you want to know more or feel interesting, please visit my poster again. And my poster number is 13. And you also can go online because my work is already on the biorecord. And thanks for your listening.
The last presentation will be by Jianto Zhang, who was a trainee at the time of the submission, but is now, within a month, a research investigator. Congratulations. He is in the department. Sorry to embarrass you. This is the love that we're talking about, reducing the stress. He's in the Department of Molecular Integrative Physiology in the laboratory of Liang-Yu Ru. And the title of his presentation is RNA Binding Protein, YTH-DC1, critically regulates liver metabolism and homeostasis.
Thank you, Dr. Sim, for the introduction, and thank you for the wonderful symposium. So today, I'm very excited to share our recent findings about how RNA modifications, specifically M6A modification and its reader protein YTH-DC1 in regulating liver metabolism. M6A is, let me try this. M6A is one of the most abundant RNA modifications that play a key role in RNA processing, including splicing, stability, and translation. This modification is installed by this metal-3 metal-14 complex and is interpreted by its RNA reader proteins, including YTH-DC1. M6A has been extensively studied in the development and cancer research, while its role in the metabolic regulation, particularly in the liver glucose and lipid metabolism, remains largely unexplored. In my previous work, we have found that this metal-3 metal-14 receptor complex are upregulated in the obese condition in the liver, which promoting M6A methylation on this G6PC RNA, which is a key gluconeogenic gene, increases the liver hepatic glucose production, contributes to the diabetes development. So this led us to the next question: how YTH-DC1, a major M6A reader protein, regulates liver metabolism. To explore this, we generate. We generate this. We first screen these M6A readers, and we found that this YTH-DC1 is very dramatically decreased in the mast cell condition, which is the metabolic dysfunction associated steatosis liver disease. And then we speculate that this YTH-DC1 is a very major player in this steatosis liver disease. And then we generate this YTGC1 liver-specific knockout mice. And very strikingly, we found that even under normal choke condition, both male and female knockouts develop severe fatty liver, including steatosis and liver inflammation and injury. So mechanistically, we found that YTGDC1 can directly bind to CD36 and suppress its expression. And in the YTGDC1 deficient livers, CD36 level expression increased a lot and drives the increased lipid uptake and promotes the fatty liver disease. We further confirmed this by restoring CD36 level to the normal condition by using this AAV shRNA. And we found that by bringing the CD36 level to the normal level in the YTGDC1 deficient liver can very significantly rescue the liver disease phenotype. So in conclusion, my study highlights YTGDC1 as a critical post-transcription regulator in the fatty liver disease by suppressing CD36 level, CD36 mediated lipid uptake and protect the liver from developing fatty liver disease. Thank you.
I'm not going to even move this thing, sorry. So that wraps up the lightning talks. I just want everyone to give a big round of applause to our speakers who are phenomenal. And their mentors who I hope are here and very proud of them because I think we are all very proud of them. This is our future scientists in these posters and giving these talks. Thank you.
We're ready to get started for our final session of the day. We are looking forward to 3 more speakers, and Dr. Martin Myers will be introducing them. So thank you.
So our first speaker for this session is Dr. Lauren Oshman. It's a real actually privileged to introduce Lauren because she's actually a neighbor as well as a family physician who has a lot of interest in type 2 diabetes and obesity. And although she didn't list it here, she's also one of the most important leads in our weight navigation program. So, Lauren.
Well, I was going to start by saying thank you, but I'm going to start by saying that my dog does not like Martin's dogs. And Martin's dogs are lovely. They're really some of the nicest dogs you could hope to meet. So my life goal is to have a dog who participates with other dogs. But I do want to thank Dr. Runge and all of the organizers for inviting me here today, particularly because today we're focusing on bench to bedside and really considering primary care, not even hospital medicine, as the bedside where we're meeting our patients. I think, again, just very, very appreciative of bringing us to that last mile problem in treating obesity. So one of the hats that I do wear that's going to be important in this talk is as a weight navigation physician. So I see patients as a consultant, board certified in obesity medicine in our clinic in Chelsea. So I'm going to start by framing this talk around the experience of one of my patients. And so I'll tell you the story of Diane. She's 42, she has hypothyroidism, she has some chronic knee and back pain that's really bothering her, and she's had obesity since her early 20s. She tried a commercial weight loss program and just wasn't able to keep up with the payments for it. And she tried other medications. She tried a phentermine topiramate, brand name Qsymia, and had side effects. It just didn't work for her. So her PCP referred her to me in the weight navigation program in 2021, and we started her on semaglutide. You can kind of appreciate her peak weight was right after losing weight from a pregnancy. And she tolerated this medicine really well. She didn't experience any side effects. She wasn't exercising, she wasn't doing resistance training, her diet was really high in processed foods, and so we built a wraparound plan around her, and she eventually was able to reach 19% body weight reduction on this medication. And she came back to see me and she said, I'm still having a lot of pain and I'd like to see if there's another option for me. And so we transitioned her to tirzepatide and she was able to lose additional weight and now is sitting at about 24% of her body weight. So I'm telling you this story because Diana's my case where everything goes right. She did really well with the medication. She's able to maintain her lifestyle and able to reach her health goals, right? Playing with her kids. So we know, and you've heard, that as little as 5% weight loss leads to substantial improvement. So as we think about how we treat populations, even our cohort of employed individuals at the University of Michigan, losing 5% of weight, reducing inflammation, et cetera, does improve your overall health for populations. But what about our individual patients, people who may benefit from additional weight loss? What about our patients who might need treatment for type 2 diabetes or other metabolic complications? Or as you've heard before, sleep apnea or osteoarthritis, compressive complications of obesity. For these patients, we have not had a lot of opportunities up until the integration of these new medications that are reliably helpful. So I'm going to talk to you a little bit about some of the older medications that we use, and you're going to see here on the top 5% body weight reduction in dark blue, and then 25% body weight reduction in light blue. And just to give you an example, phentermine topiramate, about 3/4 of patients are able to achieve about that 5% body weight reduction, and a smaller but very meaningful portion of patients are able to receive up to 10 or 15% body weight reduction. So as you heard from Dr. Griauzde, it's true that we've always had medications that work this well. But then if we can pick on tirzepatide over here in the corner, we can appreciate that 3/4 of patients are able to achieve 15% body weight reduction, and then moving even into the 20 or 25% body weight reduction, which wasn't even measured in these prior trials because it wasn't possible. So for my patients, not everyone wants to participate in bariatric surgery, which is truly the most effective option that we have right now. And there's been this frustrating gap where they've desired additional weight loss, but not have opportunities that reliably work for them. And really, this is where incretin mimetics can be helpful for us in primary care. So I'll tell you the story of how I think PCPs have become comfortable using these medications, and it really starts with the American Diabetes Association guidelines or standards of care. And in 2021, the other hat that I wear is running our state collaborative for type 2 diabetes. So we've got over 300 primary care practices across the state looking to improve guideline concordant care. So in 2021, you can see first-line therapy was metformin and comprehensive lifestyle change. And in 2022, the ADA changed their minds and they said, it depends. It depends on your comorbidities. We're gonna individualize or personalize your care for that first-line treatment. And then in 2023, we see that healthy lifestyle behaviors are still so foundational to care, but then our first-line medication is gonna depend on your medical comorbidities. And this is really where we started to see a shift in primary care physicians taking on the responsibility of prescribing these medications widely for type 2 diabetes. So this is data. Our collaborative is having its regional meetings this week, so I'll be presenting this in Troy tomorrow night. But you can see GLP prescribing in the dark blue line going up over time, and SGLT2 prescribing right underneath it increasing over time, and the green line representing the two of those added together. And so when we started the collaborative, we did a lot of education to get everyone up to speed on appropriate use of the medication, contraindications, and how to prescribe it. And I can tell you that primary care physicians are really, really comfortable doing this work because of all of the work that they do in care for diabetes. Part of this is because these medications have very few contraindications. So the endocrinologists in the room probably have patients with medullary thyroid cancer. I've still yet to have a patient with that condition or family history in my practice. And so compared to some of our older medications, there are not very many contraindications that we need to be concerned about. And this tale of two cities about tolerability that you've heard about already today. On the left, I've included some of the common side effects that you've heard about, and you've already heard that they're most common early in treatment and then tend to attenuate for many patients. And then on the right, you'll see some different clinical trials that I pulled. And these are lower dose trials from type 2 diabetes, from obesity, and you can see that in the clinical trials, the discontinuation rate for these medications was somewhere around 7 or 8%, we'll say. So this year, meta-analysis was, a larger cohort trial was conducted of 125,000 patients looking at health system data from 30 health systems, seeing what % of patients discontinued their medication. And you can see that that number for GLPs was about 65%, and for type 2 diabetes, 46%. This is very consistent with other studies that have shown somewhere between 30 to 80% discontinuation. But I'm on the promise part of my talk, not the pitfalls. And so what I take away from this is just side effects alone. Most patients can tolerate these medications. And in my practice, I would say maybe 1 out of 15 patients discontinues the medicine because of severe side effects. And what we see layered on top of this is cost, affordability, and other external issues to that patient that is leading to their discontinuation. So you've already heard a lot about how these medications confer additional benefits. And one of the things that I think is important in primary care is treating the whole person. So another drug, phentermine topiramate. Topiramate is also a really good treatment for headaches, so I might prescribe that medication. Someone who has depression, they may benefit from bupropion, so I might be prescribing them an obesity medicine that has bupropion as one of the components. But as we try to treat that whole person, I think we can really appreciate that there are many, many different comorbidities that can be accomplished and improved with these medications with incretin mimetics. So as we're trying to get more out of our medications, this class of medication has great benefit for improving the life and well-being of our patients. I think the thing that I wanted to say, because you've heard so much about this already, is that I think the other thing that these medications do, I almost think of them as a vaccine for our modern environment. And I thought about, should I put a picture of Wall-E up here? Are we becoming sedentary? Are we on our cell phones too much? Is our addictive food environment the reason that these medicines have been such blockbusters? But I think that, when we think about the promise of these medications, I do think of them as particularly useful in an environment where we do not have access to the same kind of baseline exercise activity and non-inflammatory, irritating diet that we might have had 10, 20, or even 30 years ago. And so wrapping up the promise part of my talk, I just want to conclude that primary care physicians are very comfortable with using these medications. They're easy to prescribe and titrate. They're easy to monitor and manage. And in primary care, we focus on that whole person care. We integrate the totality of someone's disease states together in a multi-morbidity model to take care of them. And we're creating a patient-centered plan that maximizes the treatment of all of their diseases while minimizing the burden and cost on them. And this is truly why I believe that obesity care has to happen in primary care. We truly know there's not specialists to refer patients to for this. And so it's why incretin mimetics are part of such an important part of our toolbox in primary care. So I've told you about the good story, what can happen when things go well. I want to tell you about some of the pitfalls that physicians that we work with have told us about. So this is work that I've participated with Dr. Kraftson, Dr. Griauzde, and others, but we surveyed physicians at Michigan Medicine a few years ago about some of the barriers that they appreciate in treating obesity care in their practices. And they told us 2 different things. Number 1, lack of training. And again, I think it goes along with this story of as treatments improve, maybe there's an additional need for new training for clinicians who treat a disease, right? And lack of time, that our physicians really are dealing with so much. My primary care physicians are my heroes. They deal with all of someone's problems in one visit. And obesity truly is such a complex problem that it deserves its own evaluation and time. You've heard from some of my colleagues, but I want to explain a little bit more about the weight navigation program just to help you understand what it is that we do. So a PCP in my practice could refer a patient to me. We do have some limitations on BMI and comorbidity just to keep demand and wait times for the program down. Before the visit, they will do laboratory testing in a variety of different questionnaires. And then we spend a whole hour with them, really understanding the story of their weight trajectory over time and how obesity has affected them, but more importantly, phenotyping them and trying to understand what are the drivers of their obesity, because we know that obesity is not homogeneous, right? One person's obesity is not the same as someone else. And then their follow-up after that visit might be with their PCP. They might need to see me again, especially if they don't tolerate a medicine we decided on or the PCP needs help with side effects, or I might navigate them to some of the treatments that we already have. So I might send them to bariatric surgery or to our weight management program in endocrinology, and then that program provides robust follow-up. So the problem we're trying to solve is really that time and training problem in primary care. And I did just want to take a moment to appreciate anyone here from UMMG, from the medical group or Caswell Diabetes Institute. When I go and talk about this program with other audiences at the American Diabetes Association or obesity medicine conferences, people are blown away that we've managed to create and invest this program in primary care in our health system. And it's just an incredibly innovative program, but more importantly, a program that can be scaled to other sites and health systems. And so I just want to thank all of you who are championing our work. I'm going to just skip past some of the information about our early program and talk a little bit about how the second pitfall affects us in primary care. And so it wouldn't be uncommon 5 years ago for a patient to have their complete physical and I've got my hand on the doorknob and I'm ready to leave and they say, "Doc, I want to talk about my weight." But I have never had someone ask me specifically for a name brand drug. Now the conversation is different. I have my hand on the doorknob and they say, "Doc, can we talk about Ozempic?" And, you know, I might have a little bit of the sweats. I've got another patient waiting to see me. This is a longer visit. But this is really where our physicians can say, "I'm going to have you go see Dr. Ashman, and she's going to do a comprehensive assessment." And so GLPs certainly are a victim of their own success, as we've seen because of drug shortages and the opening of a compounding pathway, and then patients receiving medications without the care and oversight of their personal physician. And so the point I'm trying to make is that our patients do deserve comprehensive care. And I liken this to like getting insulin, like getting your Lantus off the internet and never having anyone evaluate you or refer you to diabetes education, making sure you're not having complications, checking your A1C, your foot exam, et cetera, that it really does feel like the wild, wild west a little bit out there. And I think obesity care, because it is complex and affects all of the other healthcare conditions of patients, is best situated with their personal physician. So we ask ourselves, what is the role of the weight navigation program? What even is the role of an obesity medicine physician now in this era of new and highly effective medications? And this is all preliminary work that I'm going to show you, but I do want to share a little bit of what we found. So we looked at patients who were prescribed any anti-obesity medication, but the time frame here was after the introduction of semaglutide. And so patients on semaglutide and liraglutide for obesity are included in this, and we matched them to a propensity-matched cohort in other primary care practices. And really interestingly, we noticed that the weight navigation physicians used fewer incretin mimetics. And so this is really where the role of a specialized physician comes in, because if you don't have an experience with a lot of those other medications, you may not be as comfortable prescribing them. As I said, in type 2 diabetes, the primary care physicians are using a lot of the incretin mimetics, but maybe not a lot of these other medications. And the second thing that we found is that there was a durable difference. So even though we're prescribing fewer of this specific class of medications, our patients are on a larger part achieving 5% or more weight loss. And when prescribed these medications, our weight navigation program patients lost more weight than patients who were prescribed these same medications in primary care. And this in some ways doesn't make any sense. If two of us are prescribing the same medication, how could it be more effective from one physician versus another? And I think you heard a little bit about this in the clinical trials, where patients had, as part of the trial design, a diet and exercise program that they were expected to adhere to. But when you're getting your medicines from telehealth or maybe from your PCP, we don't have the same wraparound care to do that. And so we can appreciate, again, the medicines work better potentially when prescribed in a certain setting. And so I would say here that the context matters. Having a physician who has the team around them, the dietitian, the longitudinal remote monitoring, all of the things that we have makes these medications more successful. And I would argue that if we're going to invest money in these medications, we want to make them be as successful as possible. And so one of the clues that we have is that it does seem like patients in the weight navigation program did have more nutrition follow-up than those in primary care. And I think my primary care physicians can do this, but I will say that our healthcare system is not set up to make them succeed for this. This care program that we've developed isn't easy and seamless for all of our patients. Pitfall number 3, you've already heard a lot about cost. So I'm going to try to add something new or different to the conversation. But this is some of the internal data that you've already heard about from Dr. Runge, from our internal team at Michigan Medicine and our self-insured employee cohort. And so you can appreciate semaglutide was approved for obesity in 2021. Before that, there were 0 prescriptions, and you can see prescriptions increasing over time. And you can kind of see Saxenda, liraglutide, like getting smushed out there. You can also appreciate that I feel like the market for other medications is still there, and you can see an increasing number of patients maybe being prescribed some of our older medications, and then you can see tirzepatide kind of coming in and taking some of that market share from semaglutide in our patients. So we saw a 9-fold increase in prescribing of medications at Michigan Medicine when these drugs were approved. And so the bottom line you've already heard, and you'll hear more from Dr. Fendrick, but $300 million a year would be spent if 20% of our plan members received this treatment, and that exceeds our annual drug spend. So in the short term, how do we make this manageable for our patients when we have other medications that are less costly and more cost effective? You've already heard all about utilization management, so I'm not going to go into great detail, but as we presented in our poster, this is a really big burden on patients, on their primary care physicians and their teams, and on the health insurers. And so we may have to use step therapy and try a different medication first. We may have to do different lifestyle programs. My patients feel like, why is someone picking on me? I have to submit like logs of everything I ate and my steps and like my social security number to the insurer to get this drug approved. There may be lifetime limits. And then we saw Blue Cross Blue Shield have a class exclusion for these obesity drugs as well as Medicare. And so, I think what's not really mentioned here is the distress that it causes our patients. So the number of hours that I spent in January talking my patients through, really just dismal thoughts of like, what am I going to do? As you heard, this medicine has been so effective for me. How am I going to manage? And I think this is also really a burden on primary care teams that are already burdened, right? We already have a shortage of doctors and their teams. And as you see some of these quotes, when we asked about how they felt about loss of insurance coverage for GLP, the second quote I'll read, I sometimes feel like we've become a prior authorization factory for these medications. I would be happy to send patients elsewhere. It seems to burden the clinicians and staff significantly. And we asked them, what can we do as a health system at Michigan Medicine to help? And they said, that weight navigation program that you have, I would really love to use that. They really just need a helping hand. They feel like this is overwhelming to take care of. So just a couple last pitfalls. And I think this is important just from a physiological basis, that more weight loss is not better. So I would ask Dr. Skovronsky, I want like a Coke Light for obesity for my patients who have who are elderly or are frail or might need the anti-inflammatory effect, but not weight loss. And so I feel like it does feel a bit one size fits all with how we prescribe these medications when weight loss may not be the main issue. And then I think this last piece we've heard a little bit about from Dr. Kraftson is that we see a significant overlap and a significant number of patients with obesity have body dysmorphia. And I think what hasn't been set yet is that who decides I have a disease? Who decides that my BMI is 28 versus 26 and I have an obesity disease and I need to be prescribed a GLP-1 medication? And yet on the other hand, a pharmaceutical company needs to create a market, right? For you to see this as a problem and define it so you can have treatment. And so this piece, I do a lot of work again in fat positivity and fat bias of appreciating that there is a variety of different body shapes and none of them are better than others. I find it incredibly difficult as an obesity medicine physician to find ways to talk about this with my patients because whatever I say, they are bringing in internalized bias that affects how they see about themselves. I'm at the end of my time, even though it says 3 minutes here. So I'm going to give that to you for questions. And I'll tell you about another patient as I see the clock go over. This patient, you can appreciate 15% total body weight loss. And this patient achieved that with one of our older medications with phentermine topiramate. And so we can see whatever solution fits our patients is the one that we can use. Thank you very much.
Questions for Dr. Oshman? Well, that was fantastic. Thank you very much.
Next up, we have Dr. David Broom. Dr. Broom's a clinical assistant professor in internal medicine in the endocrine division here. And his interests are type 1 diabetes, atypical diabetes, monogenic diabetes, including MODY. And I'm just going to point out that he's one of our Caswell Diabetes Institute clinical translational research scholars. David.
So thank you so much for the opportunity to speak with you guys today. So as Martin mentioned, so just a minute here. All right, so I want to thank all of you so much for the opportunity, Martin, Dr. Runge, also Dr. Skovronsky, and everybody here at CDI, and also the Dean's Lecture Symposium for inviting me. So a brief word about this is that I'm a really big history buff, and so talking about things like podcasts that are wonderful about history, and also talking about MODY is a really, really nice piece of history ingrained here at University of Michigan. So we're going to go through that. Now, a brief word about this. So I have no relevant disclosures to this talk, but I will mention a couple of other things. So we're going to be talking about some off-label medication uses. So one of the things to know is that things like metformin for prediabetes, sulfonylureas for MODY, those are things that are not technically on label, but we do talk about them loosely all the time. Other non-relevant research, I'm the PI for a PCORI grant, which actually studies which agent is superior for SGLT2 inhibitors versus GLP-1 receptor agonists in reducing the risk of ASCVD and CKD for people. Also, I'm running a PI study for Fractal Health, which actually looks at an intervention to actually obviate the need for long-term treatment for incretin analogs for people that have obesity. So those are things that we might talk about a little bit later, or if you have questions about later, we'll talk about. So now getting into the crux of this here. So I am the PI for the long-term study of MODY monogenic diabetes here at Michigan. It's the original described pedigree of MODY here at Michigan and also across, also the longest followed. So we have more than 458 cases, and part of my job here has been actually to track down all these cases and go to Bentley Library and find out where they've been. And some of them you find, some you find that they're not living, and some people are around. And so it's a growing number of cases, so I put that plus there, sign there. And I'm also the co-chair of the Precision Diabetes Consortium. So Lou Phillipson at University of Chicago and myself, and also Tony Pollan at Maryland and Liana Billings at North Shore, we have a 10 academic institution multi-center data collaborative agreement that's actually under R01 review right now. And we have more than 4,000 people of MODY monogenic diabetes across the country that we're tracking outcomes for. So the neat thing with this is that not only are we the largest, and we're beating Exeter and also the English people, which, America, we have a very big thing for beating the English. And so I think that that's a very nice touch. So if you're ever interested, or if you have a patient you think has MODY or has MODY, just send me a message. And then I'm also part of the NIH NIDTK Rare and Atypical Diabetes Network, or RADIANT. And big shout out to Alif. I know that she's rounding right now, but she's had a very incredible role in this regard and allowed for me to tag along. And so, I'm going to talk a little bit about some Michigan's history in MODY. So we're going to go back in time for a few minutes. Also talk about the current treatments for MODY. And also, we're going to go through our data that's actually published, and you guys get a first glimpse of this. So not a lot of people have seen this. It's actually published like last week, and actually we accepted the proof this morning, so you're going to see that. All right? And we're also going to talk about what to do if you're going to give some of these new agents to people with MODY, and why it matters, okay? And then the future direction for what we're going to do with information and how we're going to actually make this the next big thing. All right, so many of you guys probably recognize this guy, Dr. Fyans. He was an endocrinologist here at University of Michigan, also coined the term of MODY and was the original person that described it, and also was the division chief in endocrinology here. So it's also a very nice guy from what I understand, although I've never personally met him, but I've seen a lot of his work over time. And so a brief word about this. In the academic year of 1949 to 1950, actually Dr. Fyans was a first-year fellow, and he was here at Michigan, and he started this long-term prospective study actually looking at the diagnosis, natural history, and clinical genetics of diabetes. And And the biggest thing to know about this is that in 1964, he uses term maturity onset type diabetes of childhood or of the young, which is like a mouthful, you know? Could you imagine saying that these days? No way. So general thinking at that time, and this is a really big thing to understand, is that there's really only two major types of diabetes, type 1 or juvenile type, which we now recognize as type 1, or maturity onset, which was now type 2, actually occurring in middle age and older people that could be controlled by diet and oral agents. And then you say, oh, it only required insulin after many years. Well, that's really interesting. So MODY, if you actually think about this, we now know it as this genetic form of diabetes that happens in young people, but it was actually originally thought to be a subtype of type 2 diabetes. And so that's an important thing to understand when you're looking at therapeutics, because many of the therapeutics actually modeled type 2 diabetes at that time. So MODY actually stands for maturity onset diabetes of the young, not youth. You know, I've been scolded many times for that. But the historic term for some forms of monogenic diabetes, okay? So monogenic meaning one gene, one type of condition, and also leading to diabetes. And it's a collection of inherited disorders of non-autoimmune diabetes. And I've listed 3 antibodies there because that's actually, you'll see this in UpToDate. That's our literature that supports you only have to check 3 antibodies to rule out type 1. And it presents at a young age. Once again, our literature, less than 25 years. And the prevalence is actually 1 to 6.5%. Now I have to do a double take almost every 5 minutes to make sure this number hasn't changed because it's actually under-recognized. So I checked like 2 weeks ago, I was like, oh my God, it went from 5 to 6.5%. Now, we still recognize that the incidence is still about 1 in 10,000 adults and 1 in 23,000 in children. And part of that discrepancy between those two is actually under recognition. And the other thing that which we're actively trying to address is that the large cohorts and studies to date have been predominantly actually, you know, Caucasians and Europeans, and they was all based in England from a very large database there. But we're actually seeking to change that actively here in the States. Now, taking a step back, a big shout out to Dr. Herman, who is my primary mentor, and he's actually responsible for demonstrating that not only is it insulin resistance, but it's actually abnormal insulin secretion that actually leads to the issue with MODY. So you actually think, you're like, oh, well, you're giving Ozempic, or you're giving tirzepatide, or you're giving one of these other agents that cause weight loss. That has to be something, right? It's the weight loss that's causing it, but it's actually the glycemic benefit. And so he was one of the original authors in 1994 that actually demonstrated that using sulfonylureas and actually looking at the insulin secretion in very elegant experiments that actually showed that abnormal insulin is actually the primary defect there. And then later, they actually found that you can give sulfonylureas, which actually cause a glucose-induced insulin secretion for patients with MODY, okay? Now, kind of going through that, sulfonylureas were then shown to be the effective first-line agents. But the downside is that we now know that if you give this to people over time, they have a decline in their beta cell reserve of about 1 to 4%. And so about a median of about 3 to 25 years, people stop responding. And then they're kind of like, well, what if I survive longer than 25 years of my diagnosis? What do you do? And that's really where adult endocrinologists start to see this issue come and where we'll talk about next, okay? A brief word about this is a nice visual from Frontiers that actually shows the primary genetic defect is actually the sulfonylurea receptor in people with the most common forms of MODY. And then there's also the glucokinase, which I've listed there, which is also the GLUT2 transporter. The thing to know about this is that the sulfonylureas actually depolarize the membrane, and that actually leads to an increase in the ATP and also increased intracellular calcium influx, which then leads to insulin release. And we'll touch on that in a little bit, okay? So sulfonylureas, we know that sulfonylureas act by actually closing the potassium ATP channels in beta cells, which leads to ATP production. And then, like we said, influx of calcium and then insulin secretion. The preclinical studies show that sulfonylurea therapy actually leads to accelerated loss of beta cell mass, which then leads to treatment failure over time. Now, looking at incretin agonists, so GLP-1s actually are known to actually activate adenylate cyclase by bypassing the genetic defect, actually. And so they lead to elevated cyclic AMP inside the islet cell, and both cyclic AMP and activated protein kinase A actually lead to generation of ATP distal to the defective channel. And then what it does is actually, since it can do that, it actually allows for insulin secretion that bypasses the genetic defect as a whole. And so what we now know is that there was a really nice study done by the Danish group, Dr. Vilsvold's group, that showed that you could actually give GLP-1 receptor agonists to patients with HNF1A MODY, which is the most common treatment requiring form of MODY, okay? But the interesting thing is that they showed there's a decrease in postprandial glucose, decrease in hypoglycemia about 10-fold, but the downside is that there was a non-statistically significant change in those values. The other thing that they didn't look at is things like weight management. They didn't look at A1C. They didn't look at sulfonylurea dose reduction. So a lot of clinical parameters left more to be desired. Now, in patients requiring additional treatment, GLP-1 has been described as add-on. And so actually, Lou and I actually wrote a paper about 5 years ago that suggested you use these agents as second-line treatment. But we'll talk about why we might actually go back on that later. Now, there was previously no clinical data, no randomized controlled trials that showed how to use this, and we're going to touch on that in just a moment. But then we published this paper in 2020, and the really neat thing is that not only was this patient population different than what we typically see, this was a group of South Indian patients. It was a father-son cohort, actually, that had HNF4 alpha MODY, and the son was actually started on Ozempic before they kind of knew what they were actually being treated for. And they had a lower A1C, it was more than 2% over time. And then checking in on him 5 years after publishing this paper, he actually had improvement in his C-peptide over time. That latter part's not published. And then the father in this cohort actually had a reduction of their A1C about 3.7%, and they were able to just stop prandial insulin completely. Then we looked at this a little bit further, and I started to get organized when I got here at Michigan and putting together a group, and we looked at tirzepatide use, or dual GIP and GLP-1 receptor agonist use in HNF1A and HNF4A MODY. So don't get too tied up on the terminology there. Those are the two most common treatment forms of MODY, okay? And what we showed is that one patient with HNF1A MODY had a 1.4% reduction in their A1C, 23% body weight reduction, and also a sulfonylurea reduction of 75%. And then we had another patient with HNF4A MODY that actually had a 3.1% A1C reduction and a 26% body weight reduction, and also the sulfonylurea was actually stopped completely. So they were able to use that just as monotherapy. Now this is just 2 cases, and so we wanted to actually partner with our buddies. So this is actually our published data in BMJ, actually, that's going to be online later this month. And what this actually showed is that this is a case series from our data, University of Cincinnati, and also University of Chicago, of all the patients that are actually receiving these agents, okay? So this is a group of patients with the 2 most common forms of MODY that are receiving treatment, and what happened before and after GLP-1 receptor agonists. And so they had an average A1C reduction of 1.3%, which was statistically significant. And then looking at the BMI reduction after receiving a GLP-1, and they had a 2.9 kilogram per meter squared BMI reduction, which actually works out to 8.9% body weight reduction in total. And then looking at their sulfonylureas, they had a 2/3 reduction in their sulfonylurea dose. So if you're taking 6 milligrams of glimepiride, you might be on 2, and you see how you do. Now, turning to our really exciting data, using tirzepatide in the same population, similar mutations, but actually just using tirzepatide, there's a 1.8% reduction. Now, the downside is that this wasn't statistically significant, and we actually think it's due to the power limitations. So we only had about 3 people in this paper that actually received tirzepatide, which isn't a whole lot. And so we actually plan to look at this a little bit further. But the BMI was really quite pronounced. There was an 8.7 kilogram per meter squared reduction in the BMI. So if you could imagine you're a BMI of 38.7 and you go down to 30 by the end of treatment, that's pretty incredible. It actually works out to about 26% body weight reduction on average. So a brief recap on this. So HNF1A MODY and HNF4A MODY, which are the two most common treatment-requiring forms of genetic diabetes from MODY. And sulfonylurea is currently recognized as first line, but we actually are actively trying to change this, because there's a high rate of treatment failure. And GLP-1 receptor agonist actually reduces A1C by 1.3%, lowers body weight by 2.9 kilos per meter squared, or 8.9%. And sulfonylurea dose is reduced by 66%. And the dual GLP-1 receptor agonist, or tirzepatide, had an A1C reduction of 1.8%, a body weight reduction of 2.9, and also 26% body weight reduction if you think about it in percentage. And every patient that was on it actually had sulfonylurea discontinued. And so the next steps, you may say, well, that's all great, but what are you going to do with this? Well, the idea is that we actually have a sequential single-arm pilot study that's under review with the grant committee currently. And we're actually looking at sulfonylurea monotherapy compared to tirzepatide monotherapy in MODY. And so the idea is that what we think is going to happen is that it'll lower A1C compared to monotherapy. It'll actually also lower body weight, hypoglycemia frequency, and reduce the risk of MACE. We're actually going to use a UKPDS risk engine calculator to predict this, because we don't expect many people to actually have a cardiovascular event. And then we're also going to look at quality of life. If you could take a once-a-week injectable agent, have lower risk of low blood sugars, what do you think you're going to have for reduced risk? The idea is that hopefully there might be a change there. And so I just want to thank a lot of people for helping make this work possible. I'm a really firm believer that no field can actually exist and prosper in isolation. You actually have to rely on a lot of your colleagues. So the Caswell Diabetes Institute has been instrumental in providing an opportunity just to even study this disease process. Also being involved with the Rare and Atypical Diabetes Network with Dr. Rao's team has been incredible. Also looking at pharmaceutical trials and the PCORI precedented trial has been a really great thing as well. And then I always put in a brief plug at the very end. If you have a MODY case, or if you suspect it, feel free to text or call me. I put my phone number up there, and just don't put it on one of those robocall lists. And then I also put my email there as well. And then another final plug, if you have any patients that don't fit the typical criteria for type 1 or type 2 diabetes, there's a link here that you can actually submit your case for review. And this is with Dr. Al's team. So I just want to say thank you so much to all of our group here. So.
Okay, do we have questions for Dr. Broom? Thank you. That was really wonderful and so interesting to hear specifically the application of GLP medications for MODY. As you talked about pancreatic burnout or lack of responsiveness to sulfonylurea, have you looked at discontinuation then of tirzepatide and whether it's a time frame issue or just the sulfonylurea causing that problem?
Yeah, so we actually, the data that show that people have a sulfonylurea discontinuation over 3 to 25 years is our own data just historically. And it's actually people that have actually burned out their pancreas, and oftentimes they do require insulin. But I'll tell you, the people that have actually you thought were burnt out and you actually give them tirzepatide, they're actually able to not be burnt out, and they're actually able to be on insulin-sparing agents. So one of the things that we made a fundamental observation that in the original diabetes care paper, the son, who was actually only like 22 years old or so when we first met him, we thought he had burnt out. He was on sulfonylurea. All of a sudden, his A1C climbed. His C-peptide level was low. He switched over to semaglutide, and he started having low blood sugars, and so they reduced his sulfonylurea. And eventually, his C-peptide came up. It was 0.6, 1.2, 2.1 5 years later. And so the thing about this is that you have what appears to be a dysfunctional islet cell, but then you're giving something that bypasses the genetic defect, and actually you're able to stimulate insulin secretion despite what's going on with the genetic defect. And so and part of that is how much do you know is coming from pro-insulin generation or insulin generation inside the islet cell. That is a whole nother discussion that I think is worthwhile exploring.
Questions?
So with sulfonylureas, over time you burn out your pancreas. Is there any evidence that that happens with semaglutide or some of these newer medications in MODY?
I think that's a really good question. I haven't seen, we haven't actually explored that question yet. Part of it is just the basis of the novelty of these new medications, but I think it's worthwhile looking into to see if there's any burnout there as well. Because who knows, if you think that only the genetic defect is in that one area, there might be actually subsequent information that shows that maybe there's also a limitation in how long you can give those agents as well. And I'm not sure that we have a good answer for that yet. Thank you.
All right, our last speaker for the day is Dr. Mark Fendrick. Dr. Fendrick's a professor of internal medicine in the Division of General Medicine. And he's going to be telling us about something that is well outside my ken, but he does these wonderful clinical and economic analyses of medical interventions like GLP-1 receptor agonists to tell us how to get maximal benefit for the most people at the least price.
Thank you. Good afternoon. I'm hopefully going to send you off with some things to think about. Didn't expect today would be a complete GLP love fest. And I hope you all heard me that that's still, in my opinion, the number one side effect of to patients now is the lack of access to them. And I'd love for you to think about how we might do that. Some of you might think that they're too expensive. Some of you might think that insurance coverage is bad. Some of you might think that clinicians aren't doing the right thing. But in my 30 plus years of practice, patients only came out of the woodwork 2 times. We spend most of our time, Lauren, right, to try to convince people to do things they don't want to do. So one was this drug called sildenafil or Viagra, where we saw men coming in who had not seen doctor for decades, and we were able to do things like screen them for cholesterol and cancer and other types of things. And the same now is these people coming in like crazy for these drugs to help them lose weight. And I just have to tell you that I'd rather not know that these effective drugs were available than know they were available and not be able to give them to the patients. And this is a pretty rare phenomenon with things that we need to all come together and work this out. So for those of you who don't know me, general internist, I spend most of my time doing applied health policy and trying to argue for patients to, as I said earlier, to enhance access and affordability. And I'm so impressed by all of the researchers from bench to bedside. I'm even on the further right of that, and want to make sure that compared to what you do in your average life and what your ambitions are and your goals are, that unlike you who publish or perish, I published and I still perished. So that's kind of important. When I made this decision 20 years ago to no longer write grants and papers, and in fact to try to start an agenda such that our patients and their clinicians, it would be easy for them to do the things for which the evidence, as we heard today, was strong. It should not be hard. It should be easy for us, we clinicians, and our patients to do things where the evidence, which is produced mostly by you all, to make that happen. So this is not unique to the area of unhealthy weight or diabetes. I think that there's never been a better time to be a patient or a doctor, or a nurse practitioner, or a PA, or a pharmacist. Like, the opportunities available to us are great. But if you go to a healthcare meeting outside of this meeting, the meetings I go to, people don't talk about health. They talk about money. That's all they talk about is money. And we have to abide by the idea, if you can't beat them, join them. We could all bang our healthy snacks and say the price of X is too high, right? But the prices of these things, whether you agree they're high or too low, there is very, very little influence of this. And if you want to be bored about the drug price negotiation programs, other types of things, but the bottom line is, just because a particular service is high price or low priced doesn't mean it's good or bad. This disconnect between how the money flows and what we are training to do to help patients and populations needs to be mended, right? The money should flow to health as opposed to the other way around. So when you think about things that we beg our patients to do, or in this rare case that patients actually come into our office saying, I've done my due diligence, I think I could really benefit for those, right? This is very problematic because just about all the things that we do, and I believe in the case of GLP-1s, are cost effective, not cost saving. And for those of you who don't live in my world and John Ayanian and others, cost effective means you have to spend money to get health. And I don't think that we should save money in healthcare. We don't do it in transportation or education or defense. I think it's worthwhile to spend. But governments and private payers now are insisting that we're cost neutral, right? And I think for those of you who know Woody Allen, the great medical researcher, he said in the 70s that the best way to reduce expenditures is death. It was true then, and it's true now. So we're not in the business of death. We're in the business of making people healthier, and we have to figure out how to spend the $5 trillion we spend on healthcare in the US every year. So the good news is just about everyone, Republicans, Democrats, management, labor, health plans, health sciences companies, they all agree that $5 trillion is enough. No one thinks it should be $6 trillion or $8 trillion. It should not be any more than 17% of our GDP, right? What we also agree on is we don't get the health benefits that we should compared to other countries that spend far less, right? So we need to move the money around. That is what I've been doing for 30 plus years. I need your help to move the money around. Very sophisticated academic work, which is more of the good stuff, which I believe, I'm convinced from today, that is GLP-1s, incretin mimetics, and other types of things, and less on the things that we're spending billions on that at best don't make Americans any healthier, and in many situations cause harm, right? So instead of saying, like in the Washington Post last week, that all this weight loss will lead to all these things, fewer heart attacks, fewer knee replacements, all these other types of things that take years to incur, we need to finally start focusing on those things that we buy because we can, and because incentives in our place for our clinical colleagues for us to do just because we can. And that has really been the crux of our work. And you could see how this will tie in. When I learned about this class of drugs about 5 years ago, payers turned to me and said, how are we going to deal with this? Like, we think they're too expensive. There are going to be people like you who say they're high value and cost effective. And as my colleague Mike Chernu says for 20 years, that if you buy more things that don't save money, you will not save money, right? Even though they are cost effective and high value. So how do we ultimately pay for this? If I were to ask you, which I won't because I'm being blinded by the spotlights, how many of you think we should have greater access to these drugs, the incretin mimetics, right? I know you all feel that. How many of you think they're too expensive? So we should just go off to the cocktail hour and say that's great and agree, and we're not going to get anywhere. So we have to figure out a solution, I hope, that will make everyone a little happier and everyone a little unhappier, because that's the only way that we're going to make this happen. So you've all heard about this, right? But the challenge about what's good and what's bad, and everyone is arguing, you know, when the Trump administration announced they were not going to cover these drugs in Medicare, health plan trade organizations across the country were applauding how great this was. But why? Right? Applauding that we're not allowing access to a class of drugs that you all think should be expanded, right? There needs to be some middle ground on these types of things. So what we do at our work is say, give me a pot of money, $5 trillion or $500, it doesn't matter. Tell us, let us use our skills complemented by yours to say, where can we maximize the most health for that money? In my opinion, it is not making patients jump through 25 hoops to get to a GLP-1 that Dr. Oshman would say they should probably get, nor do I think that everyone should be able to access to them for no cost to them, right? That's why this clinically driven approach is so unbelievably important. And after today, I learned even more. We could measure the broad-based clinical benefits. That's the hard part. Measuring how much we spend to achieve them, hard but easier than the clinical benefits that come with this. So it's not just GLP-1s or insulin or other types of medicines. You know, the point is that the human condition is, you know, no one-size-fits-all approach, right? I don't think that it'll be like some of my colleagues say, that GLP-1s will be like orthodontia and statins in a few years. That at one time it was only for rich people, and now everyone could get them because the prices have fallen. Is it a good thing or bad thing? Would people care about incretin mimetics if they were $5 a day? I would say most of the people who I deal with would say that's fine. I don't care if you're using it correctly or incorrectly, right? If it's not busting my budget, I'll let it go through. So acknowledging, most of you who do the real work, that it's ultimately money that's making these decisions is something that we can't just put our heads in the sand. We have to figure out a way to if you can't beat them, join them, right? So there are estimates of what people say in terms of the money we should spend on unhealthy weight, on diabetes, on mental health, on cancer, on prevention of end of life. And the bottom line is we hopefully will be a full employment act for people in IHPI to actually make some decisions, right, to say where we should spend our money based on health production as opposed to how much money is being paid by certain stakeholders. And those of you who want, you know, we have a t-shirt that says restoring health to the healthcare cost discussion. Because outside of here, we don't talk about health very much. We just talk about budgets. We just talk about deficits. We just talk about coverage or lack thereof. So you've all heard this is kind of my summary, is that the wave of demand for this class of drugs, incretin mimetics, will only grow. Even Daniel earlier, it's so new just last week, the fact that these drugs might be used to prevent dementia on top of that very, very long list that he said. Like if these drugs were really as good as they say and safe as most of you said, with the primary side effect, in my opinion, being cost, who's going to stop this demand from even growing further? I think if they work, that's a good thing. For my people and friends and colleagues on the payer side, this is a disaster. And when Lauren showed that slide that I've seen from Dr. Griauzde before, that I get a call from a journalist saying, if University of Michigan were to cover these drugs generously, we would spend more on this class alone than the entire drug spend, right? And if I would turn to you, Martin, and say, what would you say to that? I say, is that a good thing or a bad thing? If we're getting more health on just dealing with one drug class than all the others, I would invest all that money in that drug class. That's what we do. If you get more health for the dollar, then you invest in those things as opposed to finding out why the dollar should flow in a different way, because the data are the data, right? So my view of this is that we're seeing the scale of the benefits of this drug class just growing at a rapidity that I've never seen before. I open up my email and there's another condition that the GLP-1s are working for. So that's like, yes, if you make them, if you administer them, if you have more room in your clinics, you're gonna have more and more people coming for them. And what I'm seeing, although it was discussed in some detail, the cons of this class, I wanna reiterate to you that I believe for the third time, the main side effect is cost. And it's not the cost of the drug per se, it's the cost that patients who need them have to pay to get it. A very different thing. What the University of Michigan or Blue Cross Blue Shield of Michigan pays to a manufacturer like Lilly or Novo is much different than what our patients pay when Dr. Oshman says that it's appropriate for you to get this drug. Remember, Americans don't care about healthcare costs. They care about what it costs them, right? I would ask all of you, what did your COVID shot cost? You'd all say nothing, right? That's a good thing. I'm telling you, the manufacturers of the COVID shot did not accept nothing for those shots. Someone else paid. I want to figure out a way for someone else to pay, Lauren, so your patients will be able to get the treatment approach, whatever it is you say, to be low cost and accessible to them. So how much should they cost? This is rhetorical. I don't want your answer now. But as I mentioned earlier, the country's actuary, the Congressional Budget Office, said it's $35 billion for the next 10 years. And I would ask, I'm not going to ask you again, Mark, is that too much or too little? And the answer is no. The answer is it depends, right? What are we spending $35 billion on? As I mentioned earlier, we're spending probably $90 billion on things that we wouldn't buy even if they were free. So one way to get to $35 billion is to stop paying for preoperative testing on low-risk patients. Stop paying for CT scans of people with simple musculoskeletal back pain. Stop paying for PSA screening for men over the age of 70. I've been asked by this White House to calculate a thermometer of services that we should no longer pay for so we could pay for things like highly effective effective interventions. So this is a paper that was published last month led by Dina. And if you ask payers, what's the deal? They're not saying anything whether these drugs are good or bad. They're just saying they're going to break the bank, right? And if that's what they're saying, oh, what we're going to have to do is pass on higher premiums to you or higher cost sharing down the road, this is very, very problematic. So I understand your multiple positions. You want your patients and your employees to be healthy. You don't want to break the bank. You've got to count, Dr. Runge, to the regions. So here's where we are going for in terms of where the coverage issue is going. There are some people, based on the evidence that we have already, that these preventive services are so good, they should be covered at no cost under the ACA preventive services provision, right? This would obviously break the bank, make a lot of people in this room very happy, and a lot of people much healthier, right? So that's the side that's super exciting if the evidence bears out, like the rah-rah stuff we've heard today, right? But that is problematic when it comes to paying for it. At the other extreme, as Dina shows, there is not only, and Lauren did too, multiple steps that patients often have to jump through to get the drugs, even though their clinician might agree they needed it sooner, right? And what's even more problematic is, unlike something I've never seen before, is the rapid change in these policies. Blue Cross Blue Shield of Michigan made the national news by covering the not covering. The state of North Carolina went from covering to not covering. And for the first time in my career, putting it in publicly available emails saying, the reason I'm not covering is because it's too expensive. Never before is, oh, we're not sure about the long-term side effects or there's other alternatives. It was money, right? So we have to overcome that. And so you might have heard or might have seen that on their way out the door after the election, the Biden administration said Medicare could cover these drugs as legal if they want to. And just last week, it was interesting how this paper we wrote for the Trump administration to say, cover the things. We believe them to be high value, but we understand the fact that we can't afford $35 billion over 10 years. And as I said earlier, that $35 billion is likely overestimated for three reasons. One is prices come down, even if the drugs remain branded, like the hepatitis C example. Two is a key thing, which I'll talk about just for a minute. You don't need to stay on these drugs at full dose for the rest of your life to maintain weight loss. That's more of a hypothesis to you all than an actual statement of fact. And the third is to remind these policymakers that you should not continue to not cover high-value things that are expensive while you're spending a lot of money on things that you wouldn't buy even if they were free. So we wanted to switch this conversation from how much to how well. We wanted to put patients in the center and make it easier, not hard, for them to get it. And then we came up with this idea with talking with lots and lots of people. Dina Griauzde was enormously helpful in kind of helping me understand the physiology and the pharmacology and other types of things. But when people look at the trials of obesity, they tend to look at a long period of time, not understanding there's two distinct phases. One is the time that you're losing weight, active weight loss. And the other half, much, much longer, I shouldn't say half, is maintenance. So if you look from an actuarial standpoint, people lose weight on these drugs, some really quickly, not so quickly, but they often don't continue to lose weight over years. And that's maybe why I think the Michigan might have chosen the 2-year limit. I think at least that's a good excuse, Mark. I think that's a really good excuse. But the point is, I really believe, and not going to show you all the details, that for active weight loss, these drugs are fantastic compared to anything we've seen before. And I do agree with Lauren and Dina that there are some patients who should be tried multiple other things, but in the end, if they don't succeed, they should be asked and offered a GLP-1 or an incretin mimetic that they could afford. So I had some brilliant mathematicians, some at the University of Chicago, Michigan graduates, to say, what if we actually created the budget impact of separating these two things, that more people got these incretin mimetics for weight loss, and then we asked them, once they stop losing weight, to try something else to keep it off. Studies that have not been done, but fairly optimistic. The obesity medicine doctors, I know, met the association, they're really open to this. If we could get more people on it when the drug works the best, which is what I do, and substitute for something less. So we've worked on a whole bunch of things in this regard, and we've been pushing this idea of more people covered, but you stay on it for a lesser time, either the same drug at half dose or every other week, or an alternative that Dr. Oshman would say, we could try this with the important aspect of if we offer the alternative plan, they could get all these things that cost less than the retail price of the drug. The patients could get these things at no or little cost to them. They could actually share in the savings if you were created enough, if they stayed on the cheaper regimen at a lower cost to everyone, they would actually be paid a continuum. And if they started gaining their weight back, we would just put them on the drug at full dose, because every month you're off full dose, you're saving money, every single dose. So we've been modeling this and really pushing this in the current administration. Anyone else would listen. And one of the interesting things we found that if we had an alternative approach that was actually half the price of the current wholesale price for the full dose incretin mimetic, and it only worked 30% as well, meaning that you regained 70% of the weight and you lost all the benefits down the road for secondary issues, every single person who stayed on that regimen saved enough for 6 individuals to be on it for acute weight loss for a year, right? So moving the money around, which is what I've been trying to do in colorectal cancer, live birth, end of life scale, it's the same thing that's going on. If you give me a set amount of money to treat acute weight loss, we might be able to do this better. So some of you might be sitting there, I've had a lot of patients, I gotta beg them to go off these meds. They love these things, they never want them. And Dina and her team helped me do a survey of almost 600 patients who were obese, not on GLP-1s, and basically asked them, would they consider a program that would allow more people to get these meds for acute weight loss, but you had to stop using them when your weight loss plateaued, not a spending limit, not a time limit. Whenever the clinician said it's time, and in fact, you see 7 out of 8 patients said they've been more than happy to try this. Now, surveys are often misleading, we're not exactly sure, but at least like no one said, I'm never gonna consider that, right? They like the idea of this altruistic approach, particularly when supply is limited, either by manufacturing or by cost, but we're continuing to push this idea because we haven't come up with anything better moving forward. So this is just last week, despite our efforts, reaching out in any way we could, not in the peer literature, because we don't have time to do that, the Trump administration rejected that. And I sent my slides to your fabulous organizers, who made sure, because I tend to forget these things, Marshall, as you know, and I send this in thinking this is probably it. And lo and behold, 2 days later, the Trump administration, what an incredible surprise, says, oh, we changed our mind, right? So we're still not sure. So for those of you who think the idea of coverage expansion is a good idea, take heart. For the people who work with Dr. Runge, who have to worry about the budget of what might happen if Medicare covers these drugs, I'm sorry. Because, no, I know you feel that. I'm just saying someone is out there saying, oh my gosh, what are we gonna do? So I want you to all know as I wind down, this is a journey for me, our team, those of us who work outside the fabulous ivory tower in which we work, is like, it's hard ultimately to get the findings of your work into real applied policy. And I just want to say, publishing and still perishing is that thankfully we have people in IHPI, we have people in the dean's office, we have people across the university who step outside the coin of the realm of grant funding and publication to make sure that the findings that you're all putting together, as we put a package together to make an argument that more people should have access to things, the better. So unprecedented GLP-1 love fest, right? This is great. No, thanks for having me. Think about, though, those of you who are a few steps to the left of me on the idea of actually seeing your findings become truly in the bedside, applied in the real world, right? Do not just say that the drugs are too expensive, right? We're not gonna get there. We have to figure out, like, given the fact that the drugs are expensive, given the fact that you all convinced me that the drugs are terrific, we have to figure out a way within these constraints to get the drug to as many people as possible. And the key issue is the first 3 words of this last bullet of last slide is, please let it be clinically driven. All the work you're doing, from the most basic translational applied, bedside work, Lauren, the more we actually have the patient's best interest in mind, I think the better off we'll all be. So with that, and no time to spare, I thank you, Marshall, and everyone for having me. I'm gonna work as hard as I can to make it better for patients to achieve the amazing clinical benefits that come from the discoveries of places like here at Michigan Medicine. So thank you all very much.
Wow, that was great. Okay, I think we have time for a couple of questions, considering you're the last speaker. If anybody
Is this on? Hi, Dr. Fendrick. I'm Rasimjan. I'm an internal medicine resident. And for a very long time, I had zero success in getting any Ozempic or Mounjaro approved for any of my outpatients. And I was wondering if the insurance companies are simply feeding my progress note to AI and just coming up with an answer that's an automatic reject. Is this even fair? What are your thoughts?
Yeah, the question is about AI. My kids call me artificially intelligent, so I can't speak of anything related to AI. But I will say, I was talking to John Aanian, my colleague, IHPI director, that for the years that I practiced in a Taubman clinic, I'm not sure where your clinic was, but for a year, we had one person who was responsible for all prior authorization and utilization management for 8 physicians for every drug class, right? Now, one person cannot handle the prior authorizations from one busy clinician, right? So those administrative costs cost in a lot of ways to patients, to clinicians, to health plans, and everything like that. I think ultimately, the decisions that health plans are making are what you heard from other speakers, is that this is a budget impact that we cannot handle, right? This is why the plans applauded the Trump administration's decision. This is why Dina and Lauren's slide about the fact that this threat that we're gonna spend more on this drug than every other drug that you prescribe. So we have to come to terms, which is why I can't lower the price, right? I don't want to deny it for the patients that Lauren says need it the most. I do want to figure out a way to use it most effectively. So I'm curious to hear from you and others about whether we can ultimately not use the drug and commit people to it for life. Because these estimates that come from the University of Michigan pharmacy system or the Congressional Budget Office have this assumption of uptake, adherence, and duration. And Lauren already, we know about uptake and we know about adherence, but it's so interesting to me that the number that makes the actuaries and the ultimate decision makers cringe is the duration. So that's why we came up with this idea, who know where it's gonna go, is that if you just said to your insurance company that I'm gonna use it, but I'm only gonna be using it until the patient stops losing weight, you could help me, help us, help patients to see how they respond, because we'll have published soon that the budget impact of that program is about 1/3 to what other people are seeing. Because the money is in maintenance. The money is not in weight loss. Thank you. Go ahead. Sure.
Hi. So does this also work for folks who have type 2 diabetes, or is that a different pay model? Because it's the same molecule, I get it. But like, is there a different economics cost differential based on that?
So the numbers that make, and I don't mean to continue to call out Dean Runge here. The number when it comes to diabetes is not earth-shattering, and you probably know that Medicare is already covering these molecules for diabetes and other things. So that's kind of gotten them over the hump. So given the number, the true epidemiology of diabetes, you could probably correct me, 6-7% of the population, whatever, relative to 40% of the population, that's the kind of math I know how to do. From 6 to 40 is big. So I think that others could speak up. I think the coverage for people with diabetes in large private payers, as well as CMS, is quite good. I don't know what it is locally in Michigan because I tend not to play in my own backyard. So worry less, my friend, for diabetes coverage than what's happening in the anti-obesity space. No, so this is what I do. I'd like everyone else in the room who knows a lot about this area. Dina will vouch. I know very little about unhealthy weight, but I do know how when someone comes to me, we have a crisis of access and affordability. We often try to work through this to find a better way than the current one now, which is mostly in America, as Dina showed, that people don't have access to it. No one, unless you have one of these rarer indications, have access to these drugs in Medicare. right? And the idea, which was mentioned, I think, only in passing, this compounding pharmacy issue is a very, very big one. Not just the safety issues that were raised, but what ultimately comes in terms of cost of care down the road, particularly now that it's illegal, whatever that means, to fill your drugs with those folks.
Question.
I just have one question. We have this randomized controlled trial that gives an intervention to actually maintain and bring people off medications so they can sustain their. It actually has 5-year follow-up data in Germany that actually shows that it's still effective long-term. And we're doing that trial now here in Michigan with Amy and a bunch of weight management colleagues. And I wanted to ask, what are your thoughts on developing a care path in the not-so-distant future that kind of operationalizes things that bring people off of these medications or reduces a dose long-term?
So, David, where have you been all my life, number one? 2, tell the audience that you're not here. I didn't tell you to ask me that question because Dina's going to say, I asked you to ask me that. We've never met before, right? I thought, when you said Modi, I thought that was the Prime Minister of India. So it just shows you how much I know about your work. But this is exactly what Lauren and Andrew and Dina and we believe that a substantial subset of patients who sustain their weight, experts like you say it is sustained, instead of, let's say, $1,000 a month on a full dose incretin mimetic, which is what Marshall is spending, right? We for anything less than $1,000, try something different. This is the crux of the alternative weight management program, right? So let's just say it's $500. So every month they come in, their weight's the same, they're not losing any more. We save $500. We could give you, the patient, $100, and we still save $400. And when you realize that the magnitude of this issue, and again, trying to expose many of you to look at the cost of maintenance relative to the cost of weight loss, if you have that program, I want to learn more about it. What I've learned most from Dina and Andrew and Lauren is that it's not one size fits all. Some people want drugs, some people want food as medicine, some people want a package of behavioral therapy and gym memberships. What I and the actuaries care only about is it's less than $1,000. I don't care what it is, right? It could be a tarot card for David. No, seriously, for what, as long as it's less than $1,000, people are happy and not filling their meds at full dose, that's totally good for me. We're good. Thank you. Sure. No, I'd love to.
So let me thank Mark for a tremendous presentation. And I have to say, this day, even though I had to leave for the exigencies of the day for a little while, and I'm sorry I missed the lightning rounds and all, but this has been the most fun I've had for an intellectual stimulation I've had for, and I can't think of when. So, at least not at work. So I really, really appreciate all of you who've participated. I appreciate all the speakers, and just think, I really do believe that this shows so well, and Dan Skovronsky, I had a chance to speak to him a little bit later, and Gil said the same thing to me. This shows the breadth of what we do here at the University of Michigan, from the very most basic research, which is essential and changes things for, it may not be for a few years, but changes the way we look at health, to thinking about the cost of healthcare and health policy. So really an exciting day. These days don't just happen, and believe it or not, I don't just organize them. Of course, I don't do any of the organization. So I want to make a big call out to so many people who have been involved. I hate to mention any names, but I'm going to mention a couple: Carl, Martin, others who really made this all come together. And I think all of it, except for Dan's talk, will be recorded and available. I know I need to go back and listen to a couple of those first presentations and make sure I really understand the nuances around GIP, whether it's an agonist or an antagonist, or I'm looking for that answer. Anyway, but I want to thank everyone who spent their time and their efforts and their energy and their talent in making this symposium happy. happen. So we have time for a reception. And when I was walking by that food back there, though, I was just thinking, you know, this is like a obesity challenge on that table. Looks really good. And then I forgot who it was, but who talked about alcohol and food. And so I think there's going to be some alcohol too. So we'll try to round out the day in a vibrant way. But again, thank you all, and look forward to seeing some of you in the reception, and also to interacting more in the future.